Okay, started.
Okay, so we can see my presentation, right? Okay, so welcome everybody, and we're gonna start with our workshop where we're gonna discuss hard X-ray imaging techniques for biological and environmental research, what we could do before and what we will be able to do after upgrade. So in my introduction, I would go through what kind of work we were able to do for biological and environmental sciences at APS and what we can expect going forward in terms of instrumentation. So I'm gonna go through techniques which we offer. So first I want to mention X-ray fluorescence microscopy. So we have multiple beamlines which offer this capability with variety of the resolution and sample environments. And with some additional techniques available as well. So X-ray fluorescence microscopy allows high sensitivity imaging of chemical elements, like multiple chemical elements at once from the area illuminated by the focused X-rays, and then sample is rastered, scanned through the beam. So it's a scanning probe and can be time consuming depending on the size of the sample and elements you're looking at. In addition, some of the beamlines offer tachography, quite often in combination with X-ray fluorescence. Tachography offers high resolution imaging when combined with X-ray fluorescence and is used to visualize structural variations of the sample. In addition to that, we also have full field imaging in micro and nano range. So beamlines 2 bm, 7 bm, and also 13 ID offer absorption and propagation-based phase contrast tomography with laminography. 2 bm offers laminography, and TXM instrument at 32 IDB offers nano CT with Xenis capability. And additionally, we also have other combinations with other techniques like spectroscopy for better characterization of the samples. So in terms of samples, they come in different sizes from different sources. So it's nice that APS has a variety of tools covering quite large resolution range and also compatible with larger or smaller sample sizes, depends on what you're looking at. So we can go with nano imaging at bio-nanoprobe and 26 ID nanoprobe to look at in single cells and organelles within the cells. We can visualize mammalian cells and plankton. We can visualize clusters of the cells, three-dimensional cultures of the cells known as organoids, which allow people to go through multiple drug probes and so on before those studies can move on to clinical studies on the animals. And eventually humans. We can do visualization of quite large tissue sections and tissue slabs using micro-CT. And obviously for XRF, it'll be tissue section, the thinner the better. And we can go to even bigger samples like leaves or wood chunks using XRF tomography, well, not tomography, XRF, microscopy, and micro-CT. And sometimes we even expand the time scale to something which happened a long, long time ago and study fossils. Sometimes we can go as big as T-Rex, not at once though, but like at some bone fragments. So where do we get samples from? Our users bring samples from a variety of places, from up in the air, and I hope you can see my cursor. If not, it's the top left picture. So our users had been using X-ray fluorescence microscopy to study aerosol particles from different locations. We can go at the ground level and see compositions of soil aggregates and study porosity and distribution of essential elements or plant material through them. We can look at the ocean at the upper levels, looking at the plankton doing the photosynthesis, as well as at microbial cells from hydrothermal plumes. And then in addition to that, we can also every once in a while look at the bigger animals, bigger organisms from the ocean and do X-ray fluorescence tomography visualizing three-dimensional distribution of the elements. We have variety of animal samples we've been working so far with our users, starting from single cells, three-dimensional cell or two-dimensional cell cultures. We've been visualizing tissue sections from a variety of clinical applications. And we've been using X-ray fluorescence for larger samples as well as micro-CT. So here on the bottom left, we have elemental distribution through the mouse brain section using X-ray fluorescence microscopy. And on the top right, you can see a brain micro-CT tomography, which was done in collaboration with Bobby Kasturi for brain connectomics visualization for tracing neuron bodies through the slab. And we can also do time resolved at APS. We can do time resolved studies of biological samples. So Jake Soha does quite a lot of interesting work with the insects and dynamic changes within them, as well as other groups do studies like bone crack formation and propagation using micro-CT. And we also, let's move on to another kingdom, do quite a lot of work with plant samples. So we look deep in the ground at the root interfaces and uptake of either toxic elements if they are present in the contaminated soil, or in the promotion of phosphorus solubilization using endophytes. We can look, we can go up the root to the stem and branches and look at the structure of wood using XRF and micro-CT, visualize interfaces. And here is an example of imaging of the adhesive connecting to wood pieces, tomography reconstruction from sector 2B, from 2B and beamline. We can also use X-ray fluorescence microscopy and study changes in the elemental distribution in wood exposed to either different environmental conditions or fungus, like you see on the right bottom corner. And then we can go up through the plant and look at the buds of the plants, visualizing distribution of essential elements through different part of the plant. And this understanding is crucial for studies of plant fertility. So samples can be mounted in variety of the substrate depending on what the goals you have and what the resolution you're seeking for. So smaller samples like single cells, aerosol particles get mounted on silicon nitride windows or TEM grids, and those then get mounted into either specific mounts for the nanoprobes or those kind of aluminum sticks for the microprobes. If we work with larger samples like tissue sections, they're usually mounted on the stretched ultralin, which is transferred to slightly larger F-shaped mount. The Ariza box can be an option for plant studies, and then also geology sample cell, which is quite often used at sector 13, in situ cell of different variety to study sample under different environmental conditions. And in case if you want to do tomography, then some kind of tomography kinematic mount like crystallography sample support or something like that can be used. Let me cover the beamlines, which are involved, have been involved in this work over 28 years of APS operations. So we have microscopy group beamlines, which offer X-ray fluorescence microscopy tools in combination with other techniques like tachography and spectroscopy for some of them. We have nanoprobe, and we have 13 IDE with X-ray fluorescence microscopy, XRD, and micro CT, as well as 20 IDE with XRF and full field CT imaging beamlines in imaging group. So that's what we were able to see before. So what can we see now? Let's take a look. Do you see anything? I don't see anything. No one can see anything because we have no X-rays, because as you know, APS goes through the upgrade, and no one gonna see any X-rays in the next year until we go back with new beam, which will bring us new capabilities, new science. So with a new upgraded beam, we're gonna have 100 times more coherent photons than we were able to have before. We're going to have faster measurement, high resolution, higher energy X-rays for our studies. What the beamlines we can offer to biology environmental science users. I'm going to go from low resolution to high resolution. So 8BM will offer XRF microscopy as well as XRF tomography with the accommodation of larger samples. They will also have increase in their resolution. A 13 IDE, which already been offering quite a lot of nice tools like micro diffraction, XRF, XRD tomography, and spectroscopy will significantly increase their resolution using KB mirrors and will enable faster spectroscopy scans, as well as add cryogenic sample environment. 2ID microprobes are canted and fully independent. 2ID used to offer slightly larger beam than 2IDD. And 2ID had tomography option and higher flux, whereas 2IDD had microxenes and tachography capability. So we're gonna get more similar to each other after upgrade. We will have higher intensity, higher throughput, flexible sample environment, and we'll add techniques to 2ID such as tachography, laminography, and xenes. Bionanoprobe moved to 2ID beamlines as well. It was a very nice option for high resolution imaging of biological samples at cryogenic conditions with 30 nanometer resolution available. And here's one of the examples of both tachography and XRF imaging. And after upgrade, we will be able to reach 10 nanometer resolution with a few atom sensitivity and image hundreds of cells at suborganelle level. HXN, hard X-ray nanoprobe at 26, can also be used for biological and environmental science samples. It will achieve 12 nanometer resolution and expand energy range. It offers XRF and XRD. Our full field imaging beamlines will expand their capability as well. They will go fast. They will go way fast, you cannot imagine. They will go with 30 samples rotations per second at micro CT beamlines. They will enable laminography at high resolution instruments. They're going to build new instrument, projection microscope, which will allow holotomography as well, and will offer low deposition dose and will be able to accommodate thicker samples. And last but not least, we can also have TychoProbe 33ID, which also can be used for environmental science samples, offering tachography and XRF nanoprobe with sub 10 nanometer resolution. So this is a nice summary of all the beamlines which will be available after upgrade from lowest resolution to high resolution, as well as our full field imaging beamlines. So as you can see, we have quite a nice collection which may fit your research and even bring new science after BEAM is back. So we will also expect some challenges, and we're already working to deal with those challenges after BEAM will come back. So one of them, will need to improve our high performance scanning because we want to go fast. We will optimize our workflow for multi-beamline imaging because it's very beneficial for almost all the projects we are involved at. And we will also work on robust streamlining of data and analysis, as well as the management of the big data. So that's our plans for after upgrade, and we will hope to see new projects, new science, new amazing things at APS. With that, I would like to thank my group members who were involved in supporting XRF techniques, as well as other APS members involved in supporting XRF and XRD tachography and full field imaging, as well as our beautiful engineers who make all of that happen as well. And so I'm stopping now. I'm stopping sharing my screen, and I would like to move on to our speaker, Ken Kemner. So Ken, if you can start sharing your presentation. Yes, and I would like to introduce him. So Ken, he works at Argonne since 1996. He got his PhD in X-ray physics and condensed matter physics in 1993 and began using synchrotron X-rays at NSLS2 to investigate environmental science, environmental systems. Starting from 96, he developed environmental and biogeochemical research program that used X-rays from the APS. He participated in one of the first XRF imaging experiments at Sector 2 in 1997 to investigate root fungi interactions. Since then, he used more than 10 different beamlines at APS to perform numerous X-ray-based investigations of environmental and biogeochemical systems. And his talk will be about X-ray imaging of soil aggregates and sediment cores to understand environmental system functions. Please go ahead, Ken.
Okay. Can you hear me?
Yes.
Wonderful. Okay. Thank you, Olga. It's great to be with all of you virtually. Olga read the title slide, so I'll just keep moving on. One of the things, I was given a little extra time than most people to provide a little bit of a perspective and get the ball rolling. Like I said, again, it's an honor to be here, but what I thought I'd do is go back to what happened. Do we have the, are you seeing my entire slide, or have we got the video or the image? Is that better? There we go. So yeah, this is a slide that was prepared back in 1995 by the X-ray physicist as they were getting ready to turn the APS on. And they spent an afternoon thinking that maybe they'd want to start using the X-rays for environmental science, which was back in 95 was a rather novel concept. There weren't a lot of people around the world doing it, but this is the slide that they came up with back in 95. I thought I'd show it from a historical perspective. It does a good job of identifying a lot of the different types of sciences that come together to be integrated together to solve environmental science questions. You're going from the very large scale all the way down to the molecular scale. Variety of length and temporal scales, very heterogeneous in all of those. But as good as this was, the importance of oceans and microbes was something that was not thought of much back in 95. So now if we fast forward to present day, I'd say that the use of synchrotrons to understand environmental science questions and address them is much better. We've grown a lot. And interestingly, the topics that were identified back in 95 are still relevant today and in need of new insights and understanding. To prove that, point out back in 2018, we had a workshop here at the Advanced Photon Source, thinking about how the upgrade could provide new insights to different science questions in the environmental science area. And a lot of the topics that were identified by the experts in the country that were brought together for that workshop were in that cartoon I just showed. And similarly, in 2016, there was a workshop on the role of technologies in biological and environmental imaging. And a lot of those same science questions were identified in 2016 in another workshop. So with that said, the thing I'm going to focus on is the biogeochemistry of soils today. And the main question right now that our group is focusing on is understanding the biogeochemical cycling that plays a role in carbon emissions, greenhouse gas emissions, either CO2 or methane emissions into the atmosphere. And the image on the left is also from another workshop with arrows showing the perceived fluxes of carbon from the different components of the Earth system. And it's recognized, however, that the spatial hierarchy and the chemical nature of carbon within these terrestrial and aquatic environments, that's what plays the big role in the movement of carbon across these arrows. And so, with that understanding of the importance of the distribution and the chemical speciation of carbon and microbes in soils, that should give us deeper insights into how these things all work and possibly build models to better predict what's going to happen in the future. And so, first looking at the terrestrial component of the terrestrial and aquatic system, you obviously got to start with soil, being some of the most complex elements in the earth. Half the soil is usually made up of pores and interconnected cavities. They can be filled with either air or water. And the ratio of those two components has a lot to do with the types of chemistry that might be occurring, catalyzed by microorganisms in those pore spaces, which affects the movement of that carbon from the solid phase up into the atmosphere. In addition to the pore structure, you've also recognized we've got the roots and the fungal networks, organic debris and minerals all mixed together to form this physical structure, which is heterogeneous and quite complex. But again, decomposition of the carbon within this complex matrix is what determines the movement of all those arrows we showed a couple slides ago. So moving on from the physics, if you will, of the soil, there's also the chemistry that's catalyzed often by the microorganisms. That's the bio of the biogeochemistry that I'm talking about. And if we look at it very simplistically, thinking of respiration of microorganisms, either aerobic or anaerobic, where you couple a electron donor or a fuel, that would be on the left side of that graph on the right, and coupling that electron donor to an electron acceptor, going all the way down, for instance, to oxygen at the bottom right here. It has the steepest drop, meaning it's got the most energy available for respiration for the organisms, for aerobic respiration. But after that, all the other respiration, electron acceptors, all those different oxidants are all anaerobic respiration, which are quite unique with microorganisms, living structures. And a lot of those things can happen in the soils once the oxygen is depleted, either with depth as you're going down deeper into the soil because it's consumed by the aerobic organisms higher up, or it's as you're diffusing in through different soil aggregates in those pore structures. The rate of oxygen diffusion into those pores is slower than the rate of consumption by the microorganisms, creating an anoxic environment where different types of respiration processes are occurring. Maybe go back here and just point out in the middle of the diagram, there is an implicit respiration on iron oxides right about here, which is also a net energy gain for microorganisms that respire on these minerals. They evolved to do this because iron is a very, it's a major component of soils throughout the earth. And so it's readily available to organisms for a respiration process. And going then, looking at iron, shown here in the middle between iron 2, iron 3 couples, there's a variety of respirations I had mentioned earlier. The two on the bottom right here showing the iron respiration and the sulfur respiration are ones that we're particularly interested in But you can also see how there's these long bands of coupling between a particular respiration and other types of respiration. And we'll talk about the iron here. And so we, in our group, focusing on the iron and sulfur coupling processes in wet environments is something we're particularly keen in understanding. I've talked about the terrestrial, but there's also the aquatic systems in the terrestrial aquatic systems. And that's where wetlands often come into play. They're landscape features where you typically have standing water present all or part of the time in a year. There's roughly 45 million hectares of wetlands in the United States. Vast majority of them are freshwater wetlands. And they provide a variety of ecosystem services, reducing flooding, maintaining groundwater recharge during drought, etc. But all of these processes that are occurring here play a large control on the availability of nutrients, contaminants, and as I said earlier, the migration and the movement of carbon and exchange between the atmosphere and the terrestrial environment. So if you imagine a system on the left in this cartoon, where you've got the terrestrial and the aquatic environments and these interfaces where things are happening, trying to understand those systems and how it works, you can imagine an approach of doing this, which we show on the right, where you think of an elevation gradient, where you're going from where the water is on the left of that diagram and moving up in elevation to the uplands. And you imagine where those red cores are shown, where you could actually take a soil core with depth, and you would analyze with depth the distribution of the iron and the sulfur and its chemistry. Can give you some of the insight of what these microorganisms are doing with all of their anaerobic respiration processes. But looking at it with depth, you can actually go across that oxic-anoxic interface to understand what's happening at these different locations. And so one of the things we've been doing recently is actually going to the wetlands, developed the approach where we take a 3-inch or a 1-inch soil core. You can see there in the top left image. That soil core is capped, put inside of a PVC pipe with plugs on it that are flushed with nitrogen, and the soil core in that thin plastic is kept anoxic while it is driven back to the laboratory. If it's up more than 12-hour drive, we actually have coolers to keep these at a reduced temperature. So besides reducing the oxygen diffusion, we're also reducing the microbial activity rates to keep it stable at the way it was when it was collected in the field. Those cores are then brought back to the laboratory, where with depth we can extract pore water to understand what's happening with the water at those different depths. And then we can slice horizontally in direction with a large knife the soil cores. The reason for doing this is that when we're gathering the soil core, there would be smearing on the outside of that curved surface. And by putting now our thought is by putting the smearing in the horizontal direction, we can still have, we're maintaining the vertical integrity of the sample and understanding the depth profiles of the soil. Once the soil core is sliced in half like this, we can put a Kapton film around it, resealing it with a flat face, and bring that to the advanced photon source, where we can now use X-ray probes with depth in that core, as if it was still sitting in the soil back in the wetland. If we were sitting at the iron edge doing iron zanes, as depicted up here, we of course can use the zanes to depict the difference between iron 2 and iron 3 in the soil. You can then look down here in the depth profiling, where we had a 40-centimeter core, and at different depths showing what the rate, how much iron 2 there was relative to total iron 2 in the system, getting the average valence state of the iron 2. As opposed to doing just the zanes, we can do the full XFs, extended region of the spectrum, looking at the mineralogy of the iron. And these minerals tell us, again, gives us insights in the types of processes that are occurring there. And you can see how we can do a multi parameter fit of the types of minerals associated with depth in this soil core itself. All of these results then are sent off to a like a p-Flowtran model, biogeochemical model that can predict the types of respiration processes and mineralogies that might be formed in the soil under different conditions, with the hope of then being able to predict in the future what might happen at that system as it goes through wetting and drying cycles, which can be changed due to global change. In addition to looking at the iron, we can also look at the phosphorus, which is a nutrient element in the soils. Big questions are often just the state of the phosphorus. Is it organic or an organic or inorganic phase, telling you a little bit about bioavailability of the phosphorus. And where we were doing the iron measurements at sector 10, we can go just right next door to sector 9, coordinating those beam runs, so we can quickly do phosphorus measurements of those same soil samples coming from the core. In addition to the phosphorus at sector 9, we can also do the sulfur, which is now getting the coupling of those electron transfers between iron and sulfur species within the soil. And looking at those, you can see here on the left examples of a variety of standards that can be created to simulate the types of electron transfer reactions occurring in the soil, possibly either driven directly by the microorganisms or exchanged abiotically between the iron and the sulfur minerals. In addition to the chemistry, we've also trying to incorporate the understanding again of the physics and the porosities and the movement of the hydrology within these cores. And so a simple demonstration we did was to take a soil, first one was a 1-inch soil core that was collected, putting it just in a beaker of water, very simplistically at a particular height, with the bottom of the beak of the soil core open at this point, so that the water levels were going to equilibrate over a period of a few weeks in this soil core, such the assumption being that down here you'd have completely saturated soil pores, and up here, depending upon how much water wicked its way up through advection, you might still have the open pore space. And so looking at this location up here, doing a tomographic imaging at this elevated height, we were able to see a location where in fact there were still open pores, again indicated by the darkest color here, as well as water, a slighter gray color, indicating that advection of the water up in some locations. So you had this mixture of water and soil, or water and pores. We then go down to the bottom where we expected to see completely saturated pores. We found that is not necessarily what always has happened. And we could actually see a location where down here, where even a very large pore like this, there was a lot of air still in there, and you could see the rings of water on the edges of the pore itself. Could also determine organic matter, seeing that it in fact was saturated zones, and could be imaging the different plant litter that had been decomposing in that material. This was a 1-inch core. It was done at sector 7. And here we had a 1.38 micron spatial resolution. As I mentioned earlier, we were using 3-inch cores to try in the wetland studies that we do to reduce some of the smearing. And so we took another core from the Chesapeake Bay of a wetland system. You can actually see through the plexiglass here. You can actually see the wetland grasses growing at the top of this core that's collected. And looking down here in a zone which was still saturated, you can actually still see some of the water in it that was taken a few days after the tomography was collected. We took this photograph. And you can see now the inner 1-inch diameter of this core. It was imaged whole, so this is a 3-inch core, and just focused on a 1-inch slice through the middle tomographically. We can see the pores that were there. Again, we're only at 3.5 micron spatial resolution, but we can see the fact that these cores are saturated, fully saturated in this system. And the plant roots are showing up, the ones that are mineralized on the outside. And you can actually see a parenchyma formation within these plant roots that were down in that area. these soil pores that are made available for oxygen diffusion through the plant down into the anoxic zone. Interestingly, though, this particular parenchyma was fully saturated. It was not an open pore. So now that we've done the cores, we want to go into the smaller, thinking of these individual soil aggregates and provide, again, some of the historical context. Our group was the first to ever look at a soil aggregate with tomography. This is back in '98. The only reason we were first is we were just right place at the right time. Lots of work followed us and advanced us a lot further than we ever did back then. But to put in perspective what happened in '98, this 1 millimeter approximate size soil aggregate that was imaged took 3 to 4 days of data collection in full field tomography imaging. You can see a plant root that was actually running through this soil aggregate. And the data reconstruction took 6 weeks on a computer at the time. So it was not, it was a tour de force measurement back then, back in '98. Fast forwarding about 10 years, you can see the advances that were made looking at soil aggregates. Now we had data collection at that time that was less than an hour on a single soil aggregate. And the reconstruction took about a day. And now you can imagine what was exciting back then was that on the same day you collected a soil tomography data set, you could actually view it and determine if new data had to be collected on that. So this idea of feedback was something that was becoming important even back in 2008. So advancing this one slide, you'll see that I now have these little Pac-Man cartoons that I drew in here. And this is trying to connect the next big question that we're trying to address with soil aggregates, and that is the distribution of microorganisms within the soil. The problem is right now you have to draw a cartoon because the contrast, what you see with a microorganism compared to the soil pore or the water, is not very good for these very small objects, which are right at the spatial distribution or the spatial resolution you would get in a full field imaging. These organisms are about a micron in size, and again, that's about the size of a pixel. So it's still pretty hard to see something like this. So what we've done to address that is we've gone after quantum dots to use as an X-ray fluorescence tag. It's a cadmium selenide sulfide core with a zinc sulfide shell. Total size of these are about 3 nanometers in diameter. They can actually produce a optical visual color when hit due to quantum confinement of these things. And they can be surface functionalized with carboxyl or hydroxyl molecules. This surface functionalization allows us then to attach amino acids, which are a good nutrient for microorganisms. And we try to trick the organism then, you are what you eat. And the need for these amino acids, we felt would be a great way to get the quantum dot to go inside of the microorganism. To actually bring this cadmium selenide quantum dot in there and use the cadmium or the selenium as an X-ray fluorescence tag of what's in the organism. And so we did some electron microscopy to verify, in fact, that we could get these quantum dots into the cell. And we can get quite a few of them in there, such that the concentrations of the cadmium and selenium are off the charts relative to what you would expect for those elements naturally occurring in a cell. And so this image demonstrates how you can actually visualize bacteria that have quantum dots in them, these cadmium selenide quantum dots. Show you with the electron microscopy on the left that the dots light up just from a density profile. We can go to sector 2 IDD, and we were able to do X-ray fluorescence elemental mapping of those cells to see the selenium lighting up within the cell, and the optical microscopy on the right. So this is just a lot of words to describe what we did to prepare these cells. Top 3 bullets are just showing how we got the bacteria to take up the quantum dots, rinse them, get all this stuff that's not part of, inside the cell, get them off, clean them up. And then we wanted to look to see if we could see the bacterial cells colonizing soil aggregates. And so we wetted one of these soil aggregates for 24 hours to get the water to move into all the pore space. After that, we put in a small 500 microliters of these a solution containing these quantum dot labeled cells. And then we let the aggregate with the solution and these free swimming bacteria that are labeled quantum dots allowed them either 5 days or 30 days to colonize that soil aggregate. Aggregate was pulled out after 5 days, and then after 30 days, and more aggregates were pulled out. When they were pulled out, they were placed on filter paper to wick away residual medium, and then we dried them for 48 hours to have this stable aggregate to look at the distribution of the microorganisms, hopefully through X-ray fluorescence tomography. And this is what we see now, a tomographic slice of one of these soil aggregates that was collected 5 days after colonization by these cells. The green are the microorganisms from the X-ray fluorescence of the selenium. The black and the gray are the density profile of the cell in the transmission mode collected simultaneously. The red or pink colors are giving us intensity of iron X-ray fluorescence in there. And what you can see is that the majority of the microorganisms, after about 5 days, colonized and started biofilm formation on the outside surface of this soil aggregate. There were a few locations, as you can see, where a few cells had penetrated into the aggregate after 5 days, but not very many relative to what's on the outside. So getting into these micropores was not very easy in the first 5 days. But if we go to 30 days, grabbing another soil aggregate, what you see is now the complete colonization of the soil aggregate. You can actually see biofilm formation, so much concentration of bacteria in some locations that we oversaturated the detector, and you actually start seeing a yellow in the color scheme here as opposed to just the green. Very high concentrations of the bacteria. You can see this void in the middle, and if you look closely, you can see a very large number of little green dots, each one of those representing a pixel of where a microorganism would be located. It turns out that was actually another plant root that was inside of the soil aggregate. We figured that out by taking this sample over to 2BM and doing a full field image and reconstructing it. And you're looking at different cross sections in all three X, Y, and Z planes, if you will, to identify that soil root, the root that was in the soil there, and looking at, in some places, the preferential colonization relative to that root. But again, we're seeing penetration deep into the soil aggregate after the 30 days. So we now can get a rough idea of how long it takes microorganisms to swim and get into the aggregate. We also did another experiment where instead of taking the soil aggregate and dropping it and wetting it for 24 hours before adding the bacteria, we took a dried aggregate, dropped it into a solution that already had these bacterial cells, trying to simulate a rain event falling onto dry soil, where the water now you could hypothesize would be rushing in and moving into these pore spaces rather quickly, particularly relevant to the size of a microorganism. And in that case, we in fact do see that 24 hours after a soil aggregate is dropped into this solution with bacteria, you can see that the bacteria have pretty much gone into the cell, into the soil aggregate in all locations. This is just one of many slices, but we are able to find.
Hey, Ken?
Yes.
Sorry, we're kind of running out of time a bit, so.
I'm 2 slides left.
Okay, cool.
Thank you. Yeah, so you can see that they actually got into the aggregate. So in summary, microheterogeneities of environmental samples require spatial resolved analysis, their physical and chemical structure. We're developing a lot of techniques to look at these structures and the chemistry as well as the physical structure. We're using lots of different beamlines to do this. And I'll end by saying that Tomas Vargas is coming up next, and I think that some of the capabilities of this very high spatial resolution tomographic imaging that I showed of soil aggregates or soil cores, and looking at the inner 1-inch diameter of an intact 3-inch core, offers a great opportunity for collaboration and working together between what the EMSL does and what the advanced photon source does. And with that, I'm going to end with acknowledgements. I'm not going to read everybody's name off, but obviously no single person could do all the work that I'm showing here. And just want to thank everybody here for their attention. Thanks. So I will start stopping. Am I still sharing right now?
Yes, I think I can just stop you if you.
Okay, go right ahead. take it away.
Okay. Yes, so thank you, Ken. And I would like to move on to our next speaker, who is Tamas Varga. So Tamas, you can start sharing. Tamas is a scientist at the Environmental Molecular Science Laboratory at PNNL. He got his PhD in chemistry from Georgia Tech. His background is in solid state chemistry and material science, while his research today focuses on biogeochemistry and environmental sciences. He is interested in applying X-ray techniques like imaging spectroscopy and diffraction to investigate biogeochemical transformation that happen in the environment, especially in the soil. He'll be talking about correlative X-ray tomographic imaging and chemical analysis to better understand soil biogeochemical processes. Thank you, Tamas. Please go ahead.
Thank you, Olga. Can you guys hear me okay and see my slide?
Oh, yes, yes.
Thank you. So this talk will be addressing some of those you hear about cores and aggregates like in Ken's presentation. And so the challenge is, as Ken also pointed out, is to know where the microorganisms, the organic matter resides in the soil. Where are those pockets where we get stabilized? And so we're trying to get an understanding of that. And today I'm showing 3 examples that are ongoing ENVO projects that try to address these issues at kind of different scales. And the first one is going to be a kind of a bulk core scale study. Then I talk about also soil aggregates, where we look at these processes at higher resolution. And then I say a few words about our AI-based efforts. And all these, in all these cases, the X-ray portion is tomography imaging. And we try to correlate the structural data with the different chemical analyses to better understand these processes. So the first example, this core scale study, is my EMSOL intramural project with a quite large team, about a team of 10. And it is geared towards understanding the different core scale structure in different classes of soils and correlating them with the stabilization and transport of organic matter. Organic matter in this case is dissolved organic matter and mineral associated organic matter in this project. And basically we take soil cores. These are 1-inch cores to achieve about 30 micron spatial resolution from different soil classes, and first image them in the CT for the full-scale structure. Then the same intact cores go on to this column experiment, which we call stop flow. It's a hydrolytic saturated column experiment where we saturate the columns with synthetic soil water, maximize full residence time to make sure that we can extract all the water exchangeable or water extractable organic matter. And to do that by doing a series of these stop flow runs. And then we also have the, in the end, the non-exchangeable or the immobile organic matter in the soil. So we take effluent samples during these experiments for carbon, nitrogen content. We analyze the major cations that are important in soil processes, and then of course we use FTICR mass spectrometry for the chemical information on the organics. We also try to get an idea of the different soil carbon pools, how they are stabilized by mineral extractions that I will tell you about, and then we also analyze the leftover organics. And then both the structural information from tomography, the core-scale structure, and the chemical information gets put into a reactive transport model. So the idea here is in the column experiment, this kind of an overview slide of the main idea behind the organic matter composition in the pores this curve is a leaching curve that we get from the column experiment. Here the carbon concentration is plotted versus pore water volume, which is in the time axis. With different colors, we have 4 different subflow events that are carried out consecutively. And I'm also showing the sampling points with numbers on this plot to indicate where we take effluent samples. And we can see the difference between the chemical composition. These are organic groups such as saturated, unsaturated hydrocarbons, acids, lignin, proteins, and so we can, for example, look at the time point number 1. We expect that we get organics that are less tightly bound coming off from the larger pores, and they are expected to be different from those that coming off, for example, 0.6 at the end of this subflow run, where the organics from the smaller pores come out and the more stabilized organics. So these are the type of differences that we can reveal. And at this point, I only have comparison for 2 of the soil types we did. These are the arid soils type, alkali soils from Prosser, Washington, and we compared a wild and the cultivated plot. So 2 different soils in terms of their pore scale structure and organic content. So we're able to see, for example, how the average nominal oxidation rate of carbon varies between these 2 soils. And we found that in the soil that was found to be more porous, the white soil in this case, had larger porosity and more pore connectivity, had this higher nominal oxidation state of carbon. We found, for example, that there's a significant difference between the pore water composition between the 2 kinds of soils, and also as we move on into the experiment. So in the subflow experiment, seems like the protein-like and the condensed hydrocarbons are more in the smaller pores, and they are more stabilized, and they tend to come up later in subflow experiments. And then we can also compare before and after the column experiment, the organics, to get an idea of the differences between the exchangeable and non-exchangeable organics. And then I mentioned that we also look at the stabilization mechanisms of the soil carbon pools, and we do that by these mineral dissolution extractions, where we measure from the more weakly bound water extractable part all the way to the chemically bound organic matter mineral interactions or complexations. And so we can again compare this for the 2 types of soils, and we also have data from the different cations. And so we found, for example, that the cultivated soil has more carbon stabilized by absorption to crystalline minerals, while in the white soil, most of the carbon was stabilized by organic matter mineral complexation. But this shifted between the organics from before the column experiment to after. But seemed that the complexation after the column experiment was dominated by iron mineral absorption. So it is also important to correlate these findings, different carbon pools, with the time data, and so we can get an understanding of the mineral-based stabilization. So I mentioned that we do X-ray CT for the pore scale structure, and so we do that. We use that data in different ways. One is, of course, to numerically compare the pore scale structure, pore porosity, pore connectivity between the soil types. But we also able to provide these image data, the segmented data, as a kind of a library or database of soil for the user. And the third is that these the segmented 4-scale data also provides information, serves an input into reactive transport and the computational fluid dynamics model. So one way that imaging these scores at the APS could really help is the resolution. Of course, so we have about 27 micron spatial resolution with these 1-inch cores, and as we extract the probability distribution functions for information, we get an estimate of the diffusion times for the reactive transport modeling. We of course miss everything that is below, let's say 25 micron, and it's also a data issue. But if we could do some sort of region of interest imaging and stitching of the high resolution data and computationally handle that efficiently, then we could really get really important information for the models, because we know that cores below 30 microns are really important for microbial processes as well, and they of course bind a lot of organic matter. And this is just a slide to show the type of information we consider for reactive transport modeling. In addition to the structure, the 4-scale information, we also input the FTICR spec-based chemical information, so we can see how for example, the heterogeneity of the soil affects the composition of the organic matter. And so things like scaled rate and carbon use efficiency can be studied and compared between the different organic groups found and the different soils, and see how the spatial heterogeneity in the soil affects the microscopic carbon respiration rate. And so I'm moving on to the aggregate scale now, because Ken also had this distinction in his talk, where I want to highlight an internal project led by my colleague Dr. Safoku, where we try to develop a automation-ready workflow for full 3D chemical characterization of soil aggregates. And so, of course, as we mentioned, soils are very heterogeneous. You can see significant heterogeneity even in a soil core. So ideally, you would like to subsample that and look at the some of the important hotspots within that core on the aggregate scale. These are larger aggregates in this project, about a centimeter size. And this project basically uses X-ray CT, micro-CT, combined with SCM, EDX to create chemical maps. So the CT was used first to check the integrity of the soil aggregates during the treatment. So the treatment is first iodine exposure. We try to use iodine to make the organic matter more visible to X-ray, and then we do embedding in resin epoxy so that we can section the thin sections on the sample for microscopy. And so the CP verified that we did not make significant changes to the aggregate structure. So we claim that we want to impact aggregate, so we don't want to alter it too much. And we also verified that by careful segmentation of the 4-scale structure in the pristine after iodine and epoxy case to compare the 4-scale structure, which didn't change significantly. Now these are images. Yes.
Yeah, about like 2-3 minutes left.
Okay. These are images of the CT below overlaid with or correlated with the 2D microscopy maps, just to show that we're able to find the common slices. And so what's important here is that we need to do this because we want to learn about the carbon. And so it is hard with hard X-rays to learn about the chemical state of carbon. Here, on the spin sections, we can at least say something about the carbon that is hiding inside the particles. Of course, there's a strong carbon background in the resin that we can do anything with, but if we do Fourier transfer IR microscopy, TIR microscopy on the spin sections, we can learn something about the nature of the organics. And we can also see, for example, here iron in the pore space, which is probably dominates the pore space chemistry there. So we can identify and study hotspots using this information. This example is actually one that builds on this, but looks at even smaller aggregates. This is a user project by Devin Wickner, who develops machine learning aided segmentation to visualize and characterize particular organic matter in soil aggregates. So this was actually done at APS in 2BM, where also the aggregates are stained for with iodine, and we can benefit from the tunable X-ray energy of the synchrotron to do imaging below and above the iodine K edge, subtract those data, and get really the organics light up in the images. So that this is a segmentation by Devin. We can get this kind of high resolution visualization of the organic matter, which is in brown here, and then these are also areas that are thin section, and we're using FTI microscopy to learn about the organics. And then, so just to point out that using the synchrotron could really benefit a project like this. Just if you look at the scan time that we do in the microscopy in the lab, if you can do scans in an hour or minutes, and if we do something that is can give us 3D chemical information such as psychography or fluorescence tomography, then we can really do away with the sectioning. We can do other analysis in the lab for carbon, but a workflow like this could really be beneficial. The synchrotron to do imaging, chemical imaging on the same aggregate as we do the microscopy. And a final word about the AI effort that we are trying to essentially have these 2D section images, chemical maps on limited sections. We are using AI to train an algorithm to try to transfer the two-dimensional chemical maps to the CT-based 3D data, and we do that by transfer learning, training an algorithm to look at the density-based data versus the chemical maps, and ideally, in some cases at least, we hope that by doing CT and running the AI, we can get an initial chemical 3D map on the some of the store aggregates. We also use the machine learning to do AI-enabled segmentation of the porosity, which we can then upscale using AI for people trend. So basically, flow and transport process modeling. And these are just the first slides where we already segmented with AI the porosity in soil aggregate. And so next step is to bring in the chemical information on these. Take-home messages: we do need the large pores of the heterogeneous soils, of course, and the kind of the column experiment where we can learn about the bulk of the organics. But the imaging at the synchrotron will be very useful if we can do towards a higher resolution, and of course if we can do the chemical imaging on aggregates. And the AI will play a major role in both dealing with big data and also using the transfer learning, which we have 3D chemical maps at very high resolution, and we want to extend it to the 3D space. I'd like to thank my project team here, and collaborators. Without them, this wouldn't be possible, and these funding sources. And thank you very much for your attention.
Thank you, Tomasz. Do we have any questions to our speakers? If not, maybe we can take some time during the break, accumulating questions to our speakers of the first half of the session. So at this point, I'd like to move on to our next speaker, which is Georgi Babnik. So he is our scientist at Argonne. He got his PhD in molecular physiology and pharmacology from University of Chicago in 1997. He got his postdoctoral training at University of Chicago and at Argonne National Laboratory. Currently, he is a bioinformatician, molecular biologist at Biosciences Division and a senior fellow at the University of Chicago Computational Institute. He's interested in how biological systems work at cellular, subcellular, and molecular level. And his talk will be about imaging modalities and understanding the chemical exchange and physical interactions in the rhizosphere. Okay, Georgia, please go forward. Yeah.
Thank you, Olga. Yeah, good morning, everyone. So I'd like to talk about a fairly recent project that just started last October. And first of all, I'd like to acknowledge people contributing to this project. So Jessica Chase from my lab, Shi Xiaoyang, Victor, Pasha, Chung-Su, Alex from XSD, X3 Science Division, Charlene from Northwestern University, Neil Fang from DSL, Data Science Learning, and Ken from Bio, Andre Zhou, Carolina from XSD. So this project is actually focusing on the importance of and interrogation of plant-microbe interactions. So this interaction is very important for plant health, and I borrowed an image from my collaborator here, showing that a model plant, Arabidopsis, if it's grown without any microbe, has different root structure when you have the plant growth promoting rhizobacteria interaction. So you see different root morphology. And this is a very interesting interplay between the plant and microbe. So when plant would sense that there's some PGPR around, they will actually increase the exudate into the soil environment because they can basically exude up to 30–40% of their fixed carbon in the forms of amino acid sugars, organic acids, and signaling molecules. By eliciting this response with the internal signaling cascade and then in response to that, microbes will migrate and colonize the root system, and altering this root architecture will increase not only below but above ground biomass. And then that microbial colonization will help to protect the plant against pathogens, abiotic stresses, as well as it will help in nutrient mobilization. So this project is really focusing on microbe and on that plant-microbe interface, looking at these chemical and structural changes. So this was, as I said, a project funded by BCIS program. And basically what we are proposing to do is to exploit these natively colonizing microbes, their sense and response circuits, and we'll turn them into biosensor cell. So this is a collaboration between Yasuo Yoshikino's lab, Jean Greenberg's lab, UChicago, and then here in Argonne, as I said, Neil and Fang, and then from Bio, we have Jessica Chase, Rose, Ken, contributing to this project. So cells will respond to their environment and sense it, and quite often it will result in turning on pathways, and then these switches then controlled by transcription factors. They simply bind to a given ligand, and there are different ways of activating the transcription of downstream genes. And then what we do here is that we replace those downstream genes with a reporter molecule. So this is a fluorescent protein. So as you can imagine, like in a human body, for example, cells can sense basically femtomolar to millimolar range analytes. So that's a really high dynamic range that instrumentations don't currently have. I don't want to go into the details, but we do change different plant genotypes to kind of assess what's going on in this during this interaction. And then not only that, we are planning to turn on one or two of these biosensors, but also try to multiplex them. And we have three different plant systems: Arabidopsis, Camelina, and Poplar. During the previously funded project led by Ken, we developed a microfluidic setup where we could monitor plant root colonization. So these are 6 parallel channels with poplar seedlings, and we labeled the cells. Rose Wilton developed a plasmid that we could label cells stably. And then we could see not only colonization, but biofilm formation, as Ken was mentioning before, as well as different arrangements of the cells. We also developed some sensors at that point, where we could demonstrate the sensing of xylose and reactive oxygen species on the plant root, but these were not natively colonizing microbes, so we are changing that. In order to expand on the repertoire of sensor cells or natively colonizing cells, last fall we went out and for about 70 different locations collected roots, poplar roots, 2 different species, and then from that 800 isolates. And then so Jessica was able to then group these into 43 different unique polymorphologies. We did 16S sequencing to define some of these classes of microbes. And we also started to characterize them, looking at, for example, antibiotic resistance, etc. But maybe more importantly to this discussion, she also defined what carbon sources they can grow on. So on the left, I'm showing different sugars on the bottom, and then the different isolates. And so what you can see is that we will be able to identify microbes that will respond to a given analyte in that root microbe environment. For example, maltose is well sensed by these microbes listed here. Similarly, we looked at some of the amino acids and organic acids. For example, malate is well sensed by some of these organisms. So now we'll be able to turn these microbes into biosensors and then also multiplex them so they can look at that environment and sense that environment. We also, Chase in my lab was also able to label these microbes, all 43 isolates. Here are some colonization outcomes using confocal microscopy. So we find some microbes that can actually colonize all 3 plant species. And we're starting to see some of the morphologies that we observed before with the program that Ken was heading. We also looked at how we can image that root microbe interaction by staining the plant and microbe in different ways. Ken was talking about this. So here we did GFP, so that's the fluorescent protein labeled microbe as they colonize the plant, and then we use quantum dot to label the plant root. You can also use quantum dots or rhodomin dextran. You can see uptake of dextran in this case, as well as you can now multiplex and see microbes interacting with that plant root. So for the APS experiments, we prepared some samples for C and she was able to image these at the bio nanoprobe. So we had the GFP labeled cells. So these are Pseudomonas fluorescens SBW25 cells. And we also surface labeled it with gold. And then under the confocal, we made sure that they are still viable cells. And then so she was able to visualize the cells with tachography. And this is the cell outlined here, as well as we saw a strong signal here, probably aggregates of gold nanoparticles. And indeed, that was the case as she was performing the elemental analysis. I have to emphasize that I didn't have a chance to prepare the sample these properly under cryo conditions. So these were simply dried cells onto a silicon nitride window. But nevertheless, we were able to see the cells. You can see the calcium signal, for example, and then you can also see that the gold signal lit up the same area on the microbe. So suggesting or confirming that microbes are labeled with gold. So the next task is to see if we can see that root microbe interaction interface. So again, we had our cells, and in this case, we also showed that they were colonizing the poplar seedling. So now we have a poplar seedlings, a root that is labeled with quantum dot, and Ken was talking about the nature of these labelings. And what she was able to see is the, so here's the root surface. We could only, without sectioning it, so we are focusing just on the edge of that sample. And then, so she was able to identify some of the cadmium signal within that root, as well as on the surface, you can see gold label. So this suggesting that we can sort of have an ability to look at that root microbe interaction using the bionanoprobe. We also have help from Xiaoling at 8 p.m., where we simply laid down on the sample holder that Olga was demonstrating 3 root samples from 2 different plant species, Papilus tremuloides and Papilus deltoides. And within that, we are just looking at elemental distribution. We are still analyzing this data to see if we can see differences between the soil aggregates attached to those roots between the 2 species. You can see that the root structures are slightly different for both of them. And I have to mention that what I've shown you here is really part of a pilot project that Carolina and Zoe are heading. So this is, again, another part of the pilot. Since we had the root samples, the courtesy of this Eberlite pilot, we were able to look at via at the 2BM to perform micro CT. And so here's like a summary slide, and I really want to highlight here that it's a really nice setup at the Carlo beamline, and Victor was actually modifying this reporting system online while we were collecting data to help novice users such as myself here. So what you see here is that root piece section that's about 2 millimeter in height, and then the attached soil aggregates. So we're able to look at a sample that was enclosed in a pipette tip. And so we can actually follow through a larger section to see what the soil aggregation is around a root, and this is just zooming through that those Z slices there. What was interesting also that since we are able to collect root samples again for another run at 2 p.m. in December, and then while the root samples looked healthy, when we did micro-CT, you can see that the inside was already degraded. So these roots are going through death during the winter season, and new root system is formed at the tips. We also have a team, Neil and Fang. They are AI ML guided analysis group. So they've been doing a lot of correlation AI work in correlating, for example, in CT images versus MRI images for brain injury. And then they were able to use AI methods to kind of figure out if one of those CT images is actually contains information that there is a brain injury, so without using MRI. So they started to look at the soil aggregates around the root system. So this is their method, defining after thresholding some of the edges and then trying to get that segmentation going. But I think we'll have to talk to Tamás and others to enumerate the porosity as well as the connections of those pores, because as Ken pointed out to me the other day, is that its roots will basically change that porosity around them, and that's where all the activity takes place. So it will be very important for us to enumerate that computationally.
Georgie, so like running out of time.
Yeah. Oh, okay. So that's it. So this is my last slide. So we are using a DBTL type of workflow to identify some of these sensor cells, performing the tests, also bringing in APS capabilities, AI, and then refining this until we identify some really high-performing sensitive sensor cells. We also doing some automation, combining liquid handling and microscopy here in sector 84. All right, thank you for your attention. Again, I'd like to acknowledge the people who contributed to this work, as well as the funding from BER, as well as ENL LDRD. Thank you.
Thank you, Georgie. I had a quick question. So you show this nice microarray systems to grow your samples. Will any of that be available to potential users of BER, of Eberlight initiative?
Yes, that is the idea, is to be able to grow samples here, plant samples in these chambers. But I have to mention that there are other BER funded projects where they develop these chips that we also are trying to bring in. So there's so those modalities will be also available here. I think it's important to be able to grow the plants here if we want to do any APS analysis. So
yes, I think same too. Yeah, I think it'll definitely be great for our potential users and existing users as well. So thank you, Georgie, and I'd like to move to the next speaker before the break. So this is Ling Lilu. She's a professor at Zhejiang University. She got her PhD at from UC Davis in 2009, working with Professor Patrick Brown. Her main research interests are function, uptake, and translocation of metals, distribution and detoxification of metals in the plants, rhizosphere processes, and hyperaccumulation and phytoremediation. So yeah, as you can see, we are going like from the soil root interfaces and up the plants. I think we're gonna keep going like this until the end of today's session. So Lindley, please go ahead with your presentation.
Thank you. Thank you, Olga. Gonna show. Good morning, everyone. Actually, it is midnight in China. So today I want to talk about some of our work on cadmium hyperaccumulation in a very specific plant species, Psyllium. So let's just talk a little about why we did this work. As we all know that the cadmium pollution in soils has been posed serious threats to both crop production and also human health. And actually in China, maybe 7% of the soil has been reported to be contaminated with cadmium based on the field survey. So the phyto extraction, which using the plants to extract the heavy metals from the soil is very useful to remediate the polluted soil. So,
hey, sorry, Lily, to interrupt. Could you remove the message popped up on your screen? It's kind of blocked the diagram. Thank
here?
you.
Okay.
Yes, yeah, thank you.
Oh, thank you. But this technology is very strictly depending on the plants we used. We needed plants to accumulate a lot of cadmium or a lot of heavy metals in the shoots, but most of the plants is unable to do this. There's kind of very specific plant species which we call hyperaccumulator. They are capable of growing in the very high level heavy metal soils and also accumulate very high concentrations of metals in their shoots. So this can be used for the remediation of the contaminated soils. So that's why we wanted to understand why these plant species can accumulate such high levels of metals in their shoots. And the plant material we used is Psyllium. This is the cadmium and also zinc co-hyperaccumulate and also lead accumulate native to China. So it is widely used in the phytoremediation of polluted soils in China now. So our project is to understand why these plant species can hyperaccumulate the cadmium. So we used the two different ecotypes of these plant species. One is the hyperaccumulated, and the other one is the non-hyperaccumulated ecotype. So the HE type and the non-HE type. So the first we wanted to understand is that how these plant species can accumulate the cadmium. The first one we found is that this plant is very tolerant to the cadmium, and we can see that on the contaminated soils, the roots is very extensive. So the root growth can grow very well in the contaminated soils, but it's not the non-hyperaccumulated ecotype cannot. This is because the HE type has a very high SOD levels in their roots and also the antioxidant systems. And also these plant species can uptake and especially translocate a lot of cadmium to their shoots. And this is because there's very efficient cadmium transporters in the xylem and also the cadmium uptake processes, such as HMA2. These transport genes was highly expressed in the xylem loading for the xylem loading of the cadmium. But the next question is that if some such high cadmium was moved to the shoots, so why the upground tissues of these plant species can tolerate or accumulate such a high level of cadmium? So to understand these questions, we use the XIF and also the LAISV Mars to image the cadmium in the different tissues or in the upground of these plant species. And we can see after treatment of the cadmium for 7 days, and this is the HE type, which is the hypercommunity ecotype, the cadmium is highly localized in the vascular bundles, but also the pitch and cortex tissues of this ecotype. But in the non-hypercommunity ecotype, there's no cadmium here. So this means the cadmium is efficiently translocated with the vascular with the xylem from the roots to shoots. And if we extend the exposure time to the 1 month, we can see the cadmium goes to the from the vascular bundles to the cortex and also pitch, which is the cells will consist of very large vacuoles. So the cadmium was stored in these vacuoles. But in the non-hypercommunity ecotype, the cadmium was restricted to the vascular bundles. So that's the main information that we got, that cadmium in the stem of these plant species transferred gradually from the vascular bundles to the cortex and pitch tissues, and the sequestration. So finally, the cadmium concentration is higher in here than in the vascular bundles. So we think the sequestration of cadmium in these tissues of these cells is probably an active processes. Another interesting thing, we found that the cadmium distribution was very similar to the calcium distributions. So they have very similar patterns in distribution. And because we all know that calcium was sometimes was stored in the plasma cells, in the vacuoles for for signal transport. So maybe we think that cadmium was transported to the vacuoles by the calcium pathway. So the next job we did is to investigate the interactions of the cadmium with calcium pathways, and we also find that when under the stress of the calcium, the plants translocate and uptake much higher cadmium to their shoots. And also, we found very interesting that when we treated the plants with very low calcium, the cadmium was much higher in the younger leaves, which means that the cadmium may trigger the remobilization of the calcium from the old leaves to the young leaves. And this was confirmed by the XIF imaging of the cadmium. In the stems, we can see that this one is the low calcium. We can see that the cadmium signal in the vascular bundles systems was almost 10 times higher than those treated with the high calcium. And also we found that an extremely high cadmium was localized in the phloem tissues under calcium deficiency. So from these results, we can see that the calcium deficiency can trigger phloem remobilization of the calcium in this plant species. And the next question we want to ask is that if under the normal conditions of calcium, if the cadmium can remobilize within the plants of the hyperaccumulator. So we did this work. First, we found that when we move the, first we treat the plants with cadmium, and then we move to the solution without cadmium. And after 1 month, we found that significant amounts of the cadmium has been remobilized to the new growing tissues. And by XIF imaging, we found that significant calcium was localized in the phloem tissues at the junction section of the leaf and the stem. So, so we concluded that the cadmium can be easily mobilized by the phloem transport. So here to our conclusion is that this hyperaccumulator can accumulate the cadmium because it has a very enhanced root uptake and translocation, and also by the phloem mobilization from the old tissues or mutual tissues to the young tissues. So next question is that we did recently a very interesting and also by the XIF imagery, we found that this plant species can using the cadmium to against the biotic stress. So we understand why this plant species choose to hyperaccumulate cadmium in its shoots. So this is the plants with was exposed to the very high environment, very high PM powder midray environment, and without cadmium, we can see this plant was infected by the pathogen, and with the cadmium, the plants remains healthy, and also the biomass was much higher than those without cadmium. And very interesting, we also find that in this plant, in the PM affected plants, the cadmium was higher than those without the PM infection. And then we analyzed if the PM pathogen infection can induce the cadmium uptake and accumulation in this plant species. We found that if the plant leaves was affected by the PM, the pathogen, and the cadmium was much higher than the healthy leaves. And then with the help of Lucy in APS, we did the XIF imaging of these plants. This is the pathogen-affected tissue, and this is the healthy tissue. We made the XIF image of the cadmium, and also other elements. We found that the cadmium was exclusively localized to the PM affected zoo. And also we make the slides, the slides cross section of the leaf, and we found that the cadmium was mostly localized to the epidermal cells in the PM affected zoo, and we can see that the cadmium counts was hundreds times higher than in the healthy epidermal cells. And by nano XIF imaging, we found that those cadmium was mostly localized to the cell walls. So from these results, we can conclude that this plant species can accumulate a lot of the cadmium, especially under the stress of the pathogen, and to fight against the pathogen. And also we did some work on the transporters. So we found that under the pathogen infection, the transporters was highly expressed. So that's this is our results. And I want to thank all our cooperators, including Lucy and Matt in the APS. Thanks all. Thank you.
Thank you, Lingley, for a very interesting presentation. I think we had a comment from one of members of the audience. So yeah, does anyone have a question about this talk, or any questions to our other speakers of this session?
Yeah, I wanted to comment. I'm the one who left the comment in the thing. So we've overexpressed calcium exchangers in plants that localize to the vacuole. And when we do that, we see hyperaccumulation of cadmium in the vacuole. So that kind of fits with what you're showing, that, you know, calcium movement into the vacuole is predicated on movement using calcium mechanisms, right? Have you looked at the calcium exchangers on the tonoplast to see if they're actually moving the cadmium into the tonoplast that you see?
Yes, we did some work on this, but not yet finished. With the CAX transporters, we found that those the genes was highly expressed under the cadmium stress, but the function of this transporter has not been finished.
Yeah, because there's a chance, right, that the cadmium is free cadmium, or that it's chelated to something and it's moving in as a chelate, not just as cadmium, right? So could be another kind of thing too, right? Not just the caxes. I don't know.
Okay. Thank you for suggesting. Thanks a lot.
Thank you, and thank you, Lingley, for joining us at such a late time for you. Yeah, let's thank all the speakers from this first part of the session. And we will have a little break, and we will come back at 10:40 for other talks in the other half of this session. Thank you, everybody.
Thank you.
There you go, Joseph. Please go on with your presentation.
Okay, thank you very much, Olga. And it's a great pleasure to be here to talk about the work that we've been doing at the Advanced Photon Source over the past 10 years ago, and past 10 years or so. And I'm going to focus today on some variable humidity experiments that we've been doing at Sector 2 IDE using X-ray fluorescence microscopy to study diffusion through wood cell walls, and also at beamline 2BM using X-ray computer tomography to study moisture-induced swelling and shrinking within the wood cellular structure. So let's see. There we go. So in case everybody's not familiar, we're just going to briefly go over the structure of wood. And in our case, we're going to be looking at softwood. So wood is an anisotropic cellular material, where in softwood, the most common type of cell is called the tracheid, which can basically be thought of as this hollow tube, where each of these hollow tubes are cemented together used by a material called the compound quarter mineral lamella, which is actually a lignin-rich material with some hemicelluloses. And then the multi-layer cell wall has the S1, S2, and S3, which are secondary cell walls, which can be thought of as these nanofiber reinforced composites, where you have these cellulose microfibrils embedded in a matrix of hemicelluloses, which in softwood typically include glucomannan and xylan, and also lignin. And then even these microfibrils have a nanostructure. We have these elementary fibrils, which are the highly ordered cellulose, surrounded by disordered cellulose and hemicelluloses. So all these wood polymers in the amorphous parts, the amorphous cellulose, hemicelluloses, and lignin, they all have a lot of hydroxyl groups. And so they're hydrophilic polymers. And so they absorb water depending on the ambient conditions. So water in wood is measured as a function of moisture content, which is the mass of water divided by the oven dry mass of the wood. And as you go to higher humidities, you get higher moisture contents up to what's called the fiber saturation point. So what I want to point out here is that water in wood can exist in two different states. One is bound water, which is actually being held by the intermolecular attractions within those wood polymers. So that's down here in the hygroscopic regime. below about 30% moisture content. And then once you get above the 5% saturation point, then you have a maximum amount of bound water, and the remaining water forms free water, such as like liquid water inside of these empty lumina. So diffusion through wood cell walls, why is this important? So one of the major reasons is that when you think about any sort of processing step, when we're utilizing wood, such as biomass pretreatments, adhesive infiltration, when you're trying to glue two pieces of wood together, wood preservation treatments, they all involve diffusion of some sort of component into the wood cell walls. And then also, when you think about some of our durability issues in forest products, such as corrosion and fungal decay, both of these also involve diffusion of mineral ions into wood cell walls. So here's an example from an XFM study at 2IDE, where we're looking at metal fastener corrosion in copper treated wood. You can see that the iron actually diffuses into the cell walls and participates in that corrosion reaction. And then in terms of fungal decay, it's well known that fungi translocate different mineral ions into the wood cell walls. In this case, here's some work that we did showing the location of iron in wood cell walls, and in non-decayed wood, there's very little, if any, iron. And then after decay, you can see that the fungi has actually moved some iron into those wood cell walls. So we have this hypothesis that if we can better understand mineral ion diffusion in wood cell walls, then we can develop better wood protection mechanisms. So despite its importance, when you actually look at the literature, when we began this work about 10 years ago, really the mechanism for intracellular diffusion was unknown. There was a century-old assumption that there's some sort of water pathway that exists inside these cell walls, but there's no evidence that that water pathway actually existed. In terms of actually measuring how fast these mineral ions could diffuse through wood cell walls, there were a few estimates in water saturated conditions, but the estimates spanned over 5 orders of magnitude, and there was really no diffusion constant measurements at all below water saturation. And so to utilize X-ray fluorescence microscopy to study moisture effects on diffusion in wood cell walls, we built this humidity chamber that actually fits right into sector 2 IDE. Just attaches right to the kinematic mount in the beamline. And here's the fluorescence detector, and X-rays are coming from back behind here. And then the humidity goes in through this hose. And we also have a humidity and temperature sensor in there to actually measure the conditions during the experiments. And to do these experiments, we use 2 micron thick sections of late wood loblolly pine. We apply some ions locally using small drops of aqueous salt solutions. And then I'm going to show results from 2 different experiments. In one experiment, we're actually just stepping up the humidity to detect the diffusion onset. And then in the second set of experiments, we're going to do time-lapse imaging at humidity so we can acquire concentration profiles to actually measure those diffusion constants. So here's an example of our experiment to step up the humidity to detect the diffusion onset. So here's our 2 micron thick section. Here's our locally applied ions and form an aqueous solution. And then this is the region that we actually mapped inside of sector 2 IDE. For example, you can see here's the copper. Here's that ray cells that's right there. And you can see that under lower humidity conditions, we don't see any sort of mineral ion diffusion. But then once we get up above about 70% humidity, you can begin to see that there's diffusion of the copper ions through the wood cell walls. Here's another example using potassium chloride. In this case, we're in a transverse orientation. So the zinc map is a naturally occurring element in wood and is useful for seeing the different cell walls. In this case, the higher intensity zinc here, this is actually the compound middle lamella, and then this is the secondary cell wall. And again, we see that under dry and low humidity conditions, we don't see any diffusion. But then as you go to higher humidities, you see quite a bit of diffusion of both the potassium as well as the chloride. And so from these experiments that we did, for all ions tested, there's always a lower humidity threshold, which we did not see in diffusion. But then once you get up above about 60 to 90% humidity, we would see diffusion depending on some on the ion and cell wall orientation, as well as the different cell wall layers. Now moving on to our experiments where we actually measured the diffusion constants. So in these experiments, again, this is a longitudinal section where this higher intensity calcium, this is the compound middle lamella, and then on both sides here is secondary cell walls. And then we applied some, in this case, it was copper chloride. And then we kept it at about 75% relative humidity. And we actually we did some time-lapse imaging over a span of about 3 hours. And you can see that both the copper and the chloride diffused through the wood cell walls. And then we also did these experiments using potassium chloride. And then from these XFM images, we would trace the compound middle lamella and the calcium maps, because the calcium in this case was a naturally occurring element that highlighted the different cell wall layers, and then we could transfer those lines to the ions of interest that we're diffusing, and we were able to extract intensity profiles from both the compound middle lamella as well as the secondary cell wall. And then because we actually knew the section thickness, and of course we know the ion molar mass, we're able to convert these intensity profiles to concentration profiles in terms of moles per volume. And here you can see some examples for potassium chloride as well as potassium chloride, these time-dependent concentration profiles. And then we're actually able to analyze those using a simplifying assumption that the diffusion constant is a one-dimensional fixed second law in terms of non-steady state diffusion, and actually calculate the diffusion constants as a function of relative humidity, and as well as the different types of secondary cell walls for both potassium chloride as well as copper chloride. So a couple things we learned here is that, for a given salt, both the S2 and the compound core lamella diffuse at similar rates. Also for a given salt, both the cation and anion diffused at the same rates. So this is telling us that these ions are diffused through the cell wall and maintaining charge neutrality. And also the copper chloride is a bigger complex than the potassium chloride. So that helps explain why we get two different diffusion constants. So there's also a size effect, as would be expected. So these experiments were really critical to our discovery of what the actual intercell wall diffusion mechanism is in secondary in wood cell walls. That mechanism is shown here in this really simple schematic where you have your cellus microfibril and your hemicelluloses and lignin in the matrix. And what the mechanism is that as you go to higher humidities, you start to get regions where these glassy amorphous polymers the glassy amorphous polysaccharides are actually going through a moisture-induced glass transition and are becoming rubbery. And then once you get this kind of continuous rubbery phases, that's when we can actually get the long-range transport of these larger diffusants, such as mineral ions and also adhesive components through wood cell walls. So now I'm going to move on to looking at moisture swelling in the wood cellular structure. So because of the anisotropic structure of wood, also that also means that the moisture swelling is also anisotropic. So in the longitudinal orientation, you have very little swelling, whereas in the radial tangential directions, you have much higher swelling. And I like to point out that the swelling forces in wood are quite high. So the ancient Egyptians, they would actually use swelling forces to split rocks. So you could cut small grooves into a boulder and put some dry pieces of wood in there and pour water over the wood, and the swelling forces would actually be enough to actually split the rocks. And so these higher swelling forces and mismatched swelling and different wood products, they can cause things like cracks and adhesive delaminations. And just to also kind of show this, here's actually a experiment that we did at 2BM where we're just cycling between dry air and 98% relative humidity. And this is a cross ply adhesive bond line, such as like in plywood, where you see that in the longitudinal orientation you get very little swelling, whereas in this tangential orientation you get much higher swelling. And as you go through these different humidity levels, you can see that you get micro cracks and different types of deformations that are occurring inside of these wood adhesive bond lines. So despite the importance of swelling, there's still some really fundamental questions that haven't been answered that we're going to use micro XCT experiments to answer. So one of the questions is what's the role of neighboring cells in wood swelling? Like for example, does a wood cell wall next to the edge of a piece of wood swell differently than a wood cell wall in the middle? How much do wood cell walls themselves actually swell per absorbed water molecule? And so, almost all of the work studying wood swelling is done at this bulk level, where you're not actually studying the swelling in the cell wall material itself. And then also, what is the origin of the density dependent wood swelling? So it's been well known for over a century that the denser a piece of wood is, the more it actually swells. and there's been a lot of different theories for why that is, but that's still not an answered question. And so we're also going to study that. And so to study that, we're going to actually look at late wood and early wood in a piece of wood. And so in this case, early wood has thinner cell walls, so it's lower density material, whereas late wood has thicker cell walls and is a higher density piece of material. So to study swelling in the wood cellular structure, again, we built a humidity chamber at the FPL machine shop that actually fits inside of 2BM. So in this case, it's just a pretty simple capped on tube with two machine aluminum caps. And here's the hose that feeds in the humidified air from the humidified generator. And here's our sample inside of there, as well as the humidity and temperature probe to measure the conditions during the experiments. And for this experiment, we're looking at loblolly pine. And again, early wood to measure a low density wood, late wood to measure a high density wood. They measured 4 different sizes of specimens to look for size effects. So from 50 microns up to 1 millimeter. And then we were able to do these experiments at 4 different humidity levels. And these humidity levels are correlated to conditions inside of external humidity chambers. So we started out in a desiccant, where it's basically 0% humidity. So we take it out of the external chamber and run dry air through the humidity chamber, do the experiments, and then move the sample to the 33% chamber. And then a day later, we do the experiments in the XCT chamber, being conditioned at 33% relative humidity. And to do these experiments, we did a fast phase contrast micro XCT imaging, where we had scanning parameters where we could do each experiment in about 7.5 seconds. And so that helps us, like, even if there's a little bit of residual swelling and shrinking during the experiment, that we're going to minimize that effect. And then we pre-processed and reconstructed using TomoPy. And then, because we're interested in the swelling properties of the cell wall themselves, we developed a UNet convolutional neural networks for segmentation, so we can segment the air from the cell wall material itself. So here's some examples of those results. So again, to study cell wall swelling, we took these segmented binary images and along the entire stack up and down a sample, and basically we're able to calculate the cell wall swelling by just summing up the pixels that correspond to the cell wall material. And then to measure the swelling in the cellular structure, we just took those segmented binary images and did a white pixel fill. And then we took these volumes to be our cellular structure swelling. So looking at the results, so the cell walls, so these are plotted. The volumetric swelling is a function of moisture content. To get the moisture content, we did some ancillary absorption isotope experiments of both the different types of wood tissue, early wood and late wood, as well as the different sample sizes. And we saw that there was no detectable difference in moisture content for the different types of wood tissue, nor specimen size. So we're able to use the same absorption isotherm for all these experiments. And we also did not observe any sort of specimen size effect. So for both early wood and late wood, all the sample sizes are averaged together here. But what we do see a difference with is early wood versus late wood. So early wood, the less dense wood, swelled less than the late wood for the cell wall material itself. And we're able to quantify this by just fitting these to straight lines. And you can see that early wood actually swelled about 40% less than the late wood. And then because we know the amount of water going into the cell wall material, we're also able to calculate the volume change per water molecule, which was again about 40% less in the early wood than in the late wood. In terms of the cellular structure swelling, again, we didn't see a specimen size effect, but we saw that at the cellular level, both the cell wall and cell level for both early wood and late wood swelled at very similar values. And again, early wood swelled much less than the late wood. So going back to our questions here, what is the role of neighboring cells in swelling? In terms of the specimens that we tested, we didn't see any size effect observed from 50 microns up to 1 millimeter. So that's a couple of cells across up to hundreds of microns, up to hundreds of cells across. How much do wood cell walls swell per absorbed water molecule? Late wood is about 15 angstroms cubed, whereas, early wood is about 15 angstroms cubed, whereas late wood is about 23 angstroms cubed. I'd like to point out that both of these are much lower than bulk water, than a water molecule of bulk water. But this is similar to what we see for other polymers in the literature. And what's happening here is that within the amorphous polymer structure, there's some naturally occurring kind of free volume void elements, and these water molecules are going in there, and while they're still pushing out some of the while they're still pushing the polymeric structure out a little bit, they're also occupying that previously unoccupied free volume space. So that's why the volume swelling is slightly less than a bulk water molecule. And this is an important discovery because there's a lot of work in the wood literature where they're actually using this value to try to model swelling in wood structures. In terms of the origin of that density dependent wood swelling, the cell walls and lower density wood actually swell less than in the higher density wood. And we've got some pretty good ideas for why that is, but that's kind of the next question that we're working on answering. So looking forward now to the upgraded APS. Of course, like Olga mentioned at the beginning of this session, we're looking forward to be able to do faster experiments. In terms of XFM, we're kind of limited at how high of humidity we go to with how fast we could do the imaging. So it'd be great that with the upgrade APS, if we could go into higher humidities and maybe even trying to make those measurements in situ at under water saturated conditions. Also looking forward to combining XFM with dichography. A lot of the wood processing treatments, such as biorefining treatments, as well as pulping, they involve inorganic acids and stuff. And so, if we get some combined structure information, that could be really valuable for us. And of course, more samples and better statistics. In terms of XCT, going towards real-time imaging, like was mentioned earlier. I showed this before. This was time-lapse imaging. These experiments took about 12 hours. Of course, we're going to dry air 98%. But really, going forward, we're interested in doing these experiments much faster in real time. These were some experiments that we were able to do a couple months ago at 2 BM. And in this case, this is actually a piece of early wood that was put into a capillary tube that was filled with water. And we're actually able to, each one of these images, are a few seconds apart. So we're really able to actually observe things more in real time. Like here, you can see this water actually filling up this lumen, and you can see that these air bubbles coming out of the wood. So with that, I'd like to come to the end. Be happy to answer any questions. Of course, you can email me if you have any questions about anything. I'd just like to recognize all the collaborators through the years, especially at the APS up on top here at 2IDE, Sophie Charlotte and David Vine, and more recently, Evan, Lucy, and Olga. And then at the 2BM with XCT, Shenghui Zhao, and then more recently, Victor, Francesco, and Pasha. So that's what I have.
Thank you. Thank you, Joseph, for this great talk. Do we have any questions to Joseph from the audience?
I have a quick question. The talk was swell, but had to do that. But how long does it take to break that rock by the wood when you wet the wood? How long does that actually take? And then what do you think the differences are in the swelling when the plant is intact?
That's a great question. I'm not actually sure how long it'd take to break the boulder. I think we need to try that experiment at FPL. Yeah, I don't know. I'm not sure how long. In terms of swelling when the plant is intact, so in a tree, in a living tree, wood is in a water saturated environment all the time. So there's really not swelling and shrinking inside of a living tree. If the wood is being dried below the fiber saturation point in a living tree, that living tree is probably not going to be living much longer.
Okay, so yeah, thank you. How about we will move on to our next speaker. So our next speaker is Andrei Smirtenko. He is associate professor at Institute of Biological Chemistry at Washington State University. He got his PhD in 1996 from National Academy of Science in Ukraine. His research interests include mechanisms of plant cytokinesis, differentiation of plant vascular cells, phenotype intolerance to abiotic stress in plants, His work combines a variety of complex investigative procedures, including biochemical techniques, molecular biology, genetics, live cell imaging, and cell biology. Andrei, please go ahead.
Yeah, thank you very much for the introduction, Olga, and thank you very much for inviting me to give a talk at this very interesting event. Yeah, so I have only recently started using the APS just before it was shut down. So what I'm going to do is just to talk about the project in general, where it's going, and how we've used the APS and micro CT imaging so far to help to progress in this direction. And therefore my title is not exactly what we have done, but where we want to go. Yeah, so overall philosophy of this project is based on the fact of the process of plant feeding, right? So plant produce their own food using light energy in leaves, and they need carbon dioxide for it from the atmosphere. But another very important set of chemicals for this process are minerals that are taken from soil and must be transported to shoots. And of course, water is also very important because it's where all reactions take place. And so how plants do it? So they evaporate water from the leaf surface through plasmodesmata. This generates negative pressure inside the vascular system or wood, and then this negative pressure drives movement of water all the way from roots to shoots, and in trees it could be tens of meters. So it must be really high pressure and very strong cells that can survive this movement, can survive this pressure. And so another very important function of transpiration is temperature control. Like animals, right? We sweat and we evaporate water from our skin to cool us down. So when the temperature is high, plants evaporate water from leaf surface to reduce their body temperature and maintain the regular pace of chemical reactions. And which means that under high temperatures, transpiration increases, and so plants need more water to cool themselves down, and also to be able to proceed with the photosynthesis. So our group is interested in understanding how water moves under stress conditions and what restrictions does the stress impose on the water movement. So we are very interested in grasses. I'm fascinated by grasses because they are very drought resilient. So here we have lots of prairies now being converted into the wheat fields. And so the wheat is grown under dry farming conditions, which means no irrigation. And sometimes you don't have any rain for several months, and plants still can cope with it. And I find this really fascinating system to study how drought, how high temperatures affect water movements through plants, and how does it correlate with the morphology of the cells that are responsible for water movement. So as a model system in this project, we use Brachypodium distachion. It's a very close relative of wheat, and it has very similar anatomy of the water movement system. So here is a scanning electron microscopy micrograph of cross sections through Brachypodium stem. And so what you can see is there are kind of these circles, right, that look like monkey faces with eyes and then mouth and forehead. And so this structure called vascular bundles. And these are the structures responsible for movement of water from roots to shoots. If you look carefully, there are a number of vascular bundles that go around the perimeter of the stem, right? So large bundles. And if you look closer to the edge of the stem, there are a number of smaller vascular bundles. So inside, so vascular bundle is a pretty complex structure. So there are several tissues So here I have higher magnification of the vascular bundle on the left-hand side, and on the right-hand side, I mapped the different types of tissues that are located within the vascular bundle. And so you can see the main one is red, is called xylem, and xylem or wood, this is what transports water and minerals as a form of sap from roots to shoots. So you can see that xylem has two large metaxylem vessels on each side of the vascular bundle, and in between there are several smaller metaxylem cells. So in addition, here below we have a protoxylem vessel. So protoxylem forms very early during vascular bundle formation, and at the beginning, at the time, it plays very important role in supply of water and minerals to the forming organs. But then gradually their function become less important, and metaxylem that takes over the water movement. Above here, where we have monkey forehead, we have another tissue called phloem. Phloem is responsible for transporting photoassimilates that generated by photosynthesis from leaves to all other cells, other organs in the plant. And then in between phloem and then xylem, we have a layer of cells, which is called vascular fibers. These cells have very thick cell walls, and they play a structural role. So in addition, you can see that the green color is sclerenchyma cells, and sclerenchyma cells surround the vascular bundle and separated from other tissues in the stem. So very important for the water movement inside the xylem cells are these invaginations, right? You can see that inside the cell wall of the xylem cells is not smooth, right? It has kind of this pattern, and this pattern called pits. So here is a transmission electron micrograph of the vascular bundle. And here in the center, you can see phloem cells, and then here you see metaxylem vessels. And while most of the cell wall is very thick, right, and as you can see here, it appears as a dark material. So here we have between cells, part of the cell walls is very thin and much and less dense, right? And these are pits. So the pits are responsible for moving water between neighboring cells, and also through from xylem or vascular bundles to surrounding tissues. So this is kind of a model that shows what's happening with the xylem under drought stress, right? So under normal conditions, first of all, few definitions. So xylem cells consist of vessels, right? And so vessels has several cells, and you can see vessel because it's at the end of each end of the vessels they have a vessel element with bordered pits. And then in between the cells within the vessel, we have this perforated plate. So you can see that there are pits at the top of the cells, but also on the side walls, and so the water can flow between adjacent cells through pits. So when the soil moisture in decreases, then there is still transpiration from the leaf surface that creates negative pressure inside the vascular bundle, but the water supply is decreasing, and it induces formation of the cavity here. And then, if the drought continues, then cavity spreads to air cavity spreads to surrounding cells, causing embolism of the vascular bundle. So under these conditions, the flow of sap is completely blocked. And even if the soil moisture can be restored through rain, that the flow cannot be restored. And it has a detrimental impact on plant because they are not capable to continue with their normal developmental cycle. And so we are interested in understanding how peat morphology affects the spread of embolism from one cell to another. So this is just a cartoon showing morphology of a typical peat. So we have a peat membrane that's formed by a secondary cell wall. And inside we have a peat membrane that is essentially modification of the primary cell wall and the middle lamella. So it has been shown that plants that are adapted to arid climates have small pits with thick membrane, and the other way around, plants adapted to environments with high water levels, they have very large pits with thin membrane. And this is because large pits with thin membrane will facilitate water movement, and then the level of water movement or transpiration is proportional to the efficiency of photosynthesis and plant growth, which is very good. And so, which means plants must make a decision whether they'll have large pits with thin membranes, but this pits would facilitate movement of embolism from one cell to another, or they have a smaller pits with thicker membrane that will reduce the water flow and will limit the ability of plant to grow. And so our hypothesis is that plants have a mechanism to sense the environment and then can modify the pit morphology in response to environmental conditions. So this brings us to the question how plants control the pit morphology, and not much is known about it. And our goal is to understand genetics of this process. So we know that the pit morphology is controlled by microtubulus. These are nanofibers that are 25 nanometers in diameter, and they're highly dynamic. Microtubulus undergo through polymerization cycle, where they can grow, and then they can shrink. And then structural protein of microtubulus is tubulin, and then tubulin is released during the depolymerization of microtubulus and can be recycled for formation of new microtubulus. But for the sake of understanding pit development, we just must remember that microtubulus are highly dynamic and then can grow and shrink. And so what do we know so far about the pit formation? So in the precursor of the xylem cell, microtubulus are organized randomly. And then the first step in the formation of pit is breaking down this symmetry. So we have a formation of patches where in the endoplasma membrane that lack microtubulus. And so there are proteins that are responsible for this process, and these proteins induce depolymerization of microtubulus. And so what happens is, in the next step, to the border of the pits, we have recruitment of proteins that stabilize microtubulus, and they counteract activity of these proteins. And so if we have, if activity of depolymerizing proteins is higher, we have larger pits, and if we have more activity of microtubule stabilizing proteins, the pit is smaller. And so this is a tug of war between these proteins that control this process. So in our lab, we identified a protein. It's called MAP20. So this this immunogold micrograph shows that this protein accumulates at the sites of pit formation. And we also we quantified it, right? The density of gold pits is much higher at the pit, but not on the other side of the cell wall where there is no pit. And so MAP20 is a microtubule stabilizing protein. I would like to show it on these movies. So on the left-hand side movie, we colored microtubule growing ends with red color and shrinking ends with green. And you can see there's lots of dynamics going on. And on the right-hand side is a cell expressing MAP20. So there is no microtubule dynamics happening. No, there is no green or red ends. So we know that MAP20 is a very strong stabilizer. So we have shown that this protein localizes to pits, but most importantly, when we mutate MAP20, we have larger pits and the pit membrane is thinner. And so we tested the response of these plants to stress. So we subjected mutants and control plants to drought stress, and then we rewatered plants, and we looked at the percentage of survival. And so we have found that in all mutant lines we have analyzed, the survival was reduced, which we hypothesize is due to the peat size and due to the embolism, but we cannot test it because it's very difficult to image embolism in stems. And this is where we started to collaborate with PNNL on X-ray microtomography, and then we started working with APS to help in imaging the embolisms. And so
just a few seconds, a couple more minutes. Yeah.
Yes, I think you're almost done. So this is the results of X-ray microcomputed tomography, and it was really successful. So we could see cavities. So this is watered plants. This is a mild stress, and this is very strong stress. So you can see that cavitation generates black cells, and there are lots of black cells now in the stem, and this can be quantified, and we can see that under the strong drought stress, in this chart we have diameter of the cavities and their relative frequency in the stem, and you can see that at the strong drought stress we have more bigger cavities in the stem. But the problem with this technique, we cannot really see a morphology of the tissues, so we cannot tell which cells undergo cavitation, and APS helps. Okay, so I'll skip this movie for the interest of time. And APS is beautiful for this because it shows the tissue morphology and tells us where cavitation takes place. And we can also look at the frequency of cavitation events in small bundles, in large bundles, in different types of xylem cells. And it shows us really useful information about dynamics of the cavity formations and which cells are most susceptible to this process. So we also use nanotomography to look at the peat morphology, and it works, right? We can see nice peat membrane and peat chamber using this technique. And so in just conclusion, I just wanted to say that peat morphology is very important for drought resiliency, right? So we can use X-ray imaging for analyzing embolism and peat structure. And so far we have shown that metaxylem cells in large vascular bundles are more susceptible to embolism. And at the end, I would like to thank people who helped with this project. So we did work with Karen on developing of MAP20 mutants, and then in PNNL we work with people who help to grow plants, collect images and analyze, and also at APS. So thank you very much for your attention, again, for opportunity to present this data.
Thank you, Andre. Do we have any questions to Andre from the audience? If not, how about next speaker? We'll start getting ready. Our next speaker is Tracy Ponchon. She's a research associate professor at Department of Biological Sciences at Dartmouth College. She's a co-director of Dartmouth Trace Element Analysis Core and the Biomedical National Elemental Imaging Resource, where she uses ICP-MS, LAICP-MS, and LAICP-TOF. I'm not sure what it is. MS to help researchers conduct a wide range of biomedical elemental imaging projects. She has been a synchrotron user for more than 20 years, attending general user beamtime at NSLS, APS, SSRL, and NSLS too. Tracy's research focus has ranged from molecular genetics of plant metal transporters for biofortification of micronutrients such as iron and calcium, localization of arsenic in rice, and metal distribution in human placenta.
Thanks, Olga. I realized that bio was way too long, wasn't it? So,
that's okay.
I didn't know you were going to read it all out. Well, thank you for the invite to talk to you today. I'm going to start off with my acknowledgement slide just in case I get carried away. This is the group that I'm working with. You've already heard from Kendall Hershey, who's on the call today. He wins the prize for the participant who's asked the most questions so far. So Kendall's been working on the calcium transporters for at least a decade, probably more than that. Hormat is in his lab at Baylor, and I'm also working with Ryan Tapero at Brookhaven National Lab, beamline for 4BM, the XFM beamline, and then me, I'm at Dartmouth. And as Olga said, I use several elemental imaging techniques, laser ablation, and now most recently laser ablation ICP TOF to image various biological and biomedical specimens. So I want to jump right in and say, talk about calcium and why we're interested in calcium. We are interested in calcium as an abiotic stress signal. So in the cascade of cause and effect, there's an abiotic stress. This is like an environmental stress, not a pathogen, not a biotic stress. You get a calcium signal in plants. This then triggers binding with a calcium binding protein. Then you get gene expression changes, and then those gene expression changes then start affecting the distribution and abundance of other ions in the plant in response to the abiotic stress. And you can imagine that those downstream effects are meant to protect the plant or help the plant adapt to abiotic stresses. So we are not short of abiotic stresses in the world today. This is a map of sort of the billion-dollar weather events and climate disasters that happened in the US in 2022. We lost $21 billion in crop losses in 2022. And that isn't just a dollar sum. This is food insecurity. So as plant scientists, one of our most pressing tasks is to try and help plants develop to this rapid change in abiotic stresses. The one that we are going to focus on today is flooding. So this is a picture from the New York Times. This was taken in 2018, I think, 2019, just showing you the scale of the devastation when you get these periodic prolonged rain events. So this article was talking about how 19 million acres of cropland went unplanted in 2018. So what we're looking at, what we're most interested in, is flooding as an abiotic stress and calcium's role in that. But when we look at flooding as a stress, it's actually a combination of multiple other abiotic stresses. So when we think about how we're going to reproduce it in the laboratory, we have to kind of tease apart all the different components of flooding. We have low oxygen, we have low light, changes in light, we have dramatic changes in temperature, and then we have different changes in salt stress and osmosis. And all of these things are affected to different extents when we have flooding. So if you were thinking about flooding as a scenario, you have each of these components potentially at very different levels. So it becomes very difficult to recreate it in the lab. So we have to focus on one or other of these and then build up a scenario where we eventually look at how each of these stresses affects the plant's ability to deal with stress and the genes that are involved and the transporters that are involved. So what we've done, what Kendall and I and Hormat have done, is we've focused on oxygen as a stressor. We're going to start our experiments just looking at anoxia and the component of flooding where the plant doesn't have oxygen for extended periods of time. And then we'll work our way through the other stresses, and hopefully we'll understand the genetic mechanisms that can then inform breeders as to how to breed plants to be more resilient to periods of these stresses. And it's really quite simple, quite straightforward to reproduce anoxia in a laboratory setting. So you just buy these anoxia chambers and you buy these gas packs that consume all of the oxygen in the chamber, and you put your plants in there growing on agar, and then you take them to the synchrotron and you start imaging them. So this is a part of the talk where I'm going to talk sort of more about what's wrong with our experiments and how the APS upgrade could potentially help our experiments. Then I'm going to focus on what we're finding, because this is a very challenging system to image. So I want to start by saying this is our major finding, and what drove us to start imaging the plants is their tolerance. That we found that if we if we knock out 4 of these calcium transporters, the plants become very tolerant to periods of anoxia. So what you're looking at here is plates in which plants have been growing. This is a normal plant, and then this plant is lacking 1 of the CAX transporters. This plant is lacking 2 of the CAX transporters, and then this one has all 4 CAX transporters removed. And you can see by about 20 hours of anoxia, it's still doing really well. In fact, after 10 hours of anoxia, there is no effect on the plant. So you can imagine that this kind of resistance would be very useful in abiotic stress flooding. But if you think about it, what we're trying to image is challenging because it's a transient phenotype. So when we think about imaging elements in plants, we've had most of our successes imaging sort of slow, stable movement, where an element moves from A to B, such as in loading seeds with micronutrients, and that movement takes place over a matter of days. It moves from A to B and it stays at B until it's needed for some other function in the plant. What we're imaging is different. It's a fast transient movement because it's a signal. It's like a flash of light. So the element is moving from A to B and then it's going back to A again. So, and that's taking over hours or even minutes to move. So if the imaging mechanism that we're using actually takes longer to image the plant than this element is spending in point B, then we're never going to find it there. We're never going to see it there, even though it has dramatic downstream events on other elements. So just as an example of how long this movement takes in plants, this is an experiment that was done in Simon Gilroy's lab. He's going to apply glutamate to the tip of this leaf, and then he's imaging a calcium fluorophore to show you just how fast, and there's a counter here that you can see the time, just how fast this signal is moving in plants. So you see here, the second counter, it's a flush through the plant, and it seems to move like a wave. It's still, you can still see it at the end in the meristem, but we can run it again so you can see it again. The glutamate is attached to the tip, applied to the tip, and then it sort of sweeps through the plant. So that's an example of a calcium signal, and we're imaging that, but in response to an abiotic stressor, namely lack of oxygen. So there's a pattern to it, there's a localization to it, and there's movement, and there's a timing. So this is really what we need to try and image. And we've been doing this in my lab in Dartmouth, and we've been doing it at Brookhaven. And the experimental setup that we have is already very problematic, but this is really the only way that you can do this, and that would be that you would expose your wild type and your mutants to anoxia, and then you take them to the beamline where you have your anaerobic chamber, you do your exposure, and then you take a plant and you cut off the leaf and you put it onto Kapton tape, and then you image it. Now this image took 7 hours and 44 minutes, and it's a big image. And we did them side by side because we really wanted to have that comparison without having to quantify individual maps. And what you can see is, and this is under normal aerobic conditions, you can see that the mutant that doesn't have 4 of the CAX transporters already has a very contrasting elemental distribution. All of this calcium that is in the leaf hairs doesn't occur in wild type plants. So we've got some nice data, but what I want to dwell on here is the 7 hours is still too long for us to image this effect. Plus, we're imaging something that might be very easily might be completely perturbed by the fact that we've just plucked the leaf from the plant. So if we're measuring abiotic stress and calcium signals in response to abiotic stress, we don't want to give it additional abiotic stressor to deal with, which almost surely taking off the leaf and then imaging that leaf leaf is dying, it's undergoing abiotic stress, and we're trying to measure, capture cascades of elements that in response to anoxia when we're measuring now something else. So this is what we're left with doing. Plus, it's a different plant every time. We obviously have to image each individual plant. So we've got to find the exact same leaf of that, in that floret of Arabidopsis, we've got to find that every time and image that exact same leaf. Another artifact with these experiments, another problem that we're having is just the time period required to taking that plant, cutting off the leaf, sticking it on Kapton, going, shutting the hutch, setting up the coordinates of the map. It's already too long. There's already cascades of biological effects that are happening that we are going to start imaging that we don't mean to image. So what we really want to do, and this is just a diagram of what we really need to do, is pick the same leaf each time in this experimental setup. So you can see here, Arabidopsis leaves grow in a spiral. So leaf 1 and leaf 2 are here, then 3, 4, then 5. So when we come to the synchrotron, we have these 14-day-old plants. So they only have a total of 7 leaves, only about up to leaf 4 we can actually see. So we're there, we have to be as quick as we can, get those leaves onto Kapton tip so we can image them. If we pick the wrong one, this is an example of what happens. It has a completely different chemistry. It doesn't have the cotyledons of these first 2 leaves here. They don't have any leaf hairs at all, which is incredibly important to the calcium regulation in this plant, or so it appears. So they have a completely different distribution of calcium. So we really need to completely change the way we image if we are going to image these fast transient phenotypes that are kind of characteristic of living things. So let's just take a moment to see what we've actually found with the imaging that we've done at Brookhaven. These are the leaves that we've imaged post after 8 hours of anoxia. So you've got normal, and then you've got just a plant, the same plant that's been exposed to anoxia. You can see there's definitely some calcium movement. You can see a lot of movement in chloride. Chloride is interesting. It's involved in turgor pressure because it's, salts, sodium chloride. So we can image sort of wilting happening by just looking at the change in the distribution of chlorine. We do see changes in manganese, and we do see changes in zinc. They're not showing up very well in this map, but we see in zinc, we see that it's localized around the stomatal pores, so the guard cells around the stomatal pores, which makes sense because when that plant is exposed to a lack of oxygen, it's going to open the stomatal pores to try and get more oxygen inside the leaf. So we do see lots of changes, but we've had to really try and streamline the organization of this because there's so many elements involved, and there's the normal progression of anoxia, and then there's the mutant progression of anoxia. So we are really struggling to image fast enough to capture what's going on. Now this is the again in detail, this is the big image that we're seeing of the wild type and the mutant. So we know that that there is a difference in the baseline distribution of elements in these two. We decided to try laser ablation ICP-MS because this allowed us to image 1 hour 40 minutes per leaf, as opposed to sort of 2 hours or 3 hours at the synchrotron. So we did take definitely a hit in the resolution, but what we were able to do with laser ablation ICP-MS was just speed up how many leaves we could image over what period of time. So this experiment involved me getting up at 3 o'clock in the morning, exposing these plants to an anoxic condition, and then getting them out of that anoxic condition when I got to work, and switching on the laser and running them every 2 hours. And this gave us some quite tantalizing results in that we did see, and this is 3 different mutants. Again, this is missing 1 CAX gene, and this is missing all 4 CAX genes. And we did start to see, and if you remember the video of the pulsing of calcium moving, we do start to see some indication that we've just missed waves of calcium, like here, also at the base here. And then these 2 also show that calcium is kind of waved through the leaf. Again, that's a complete arm waving. Unless we can image faster and more often, and we're never going to catch this happening. The interesting thing that we're seeing down here in the mutant is nothing moves. Nothing gets the calcium that we see enriching these leaf hairs, it never gets out. And that seems reasonable because we've taken away all of the CAX transporters that it would ordinarily use to mobilize calcium out and send that signal that it's under abiotic stress. So we still have some quite tantalizing results. Another thing, we've done so much imaging at Brookhaven, I decided to take all of our elemental maps and put them together in a time series and try and sort of make sense out of them. And I think all that we can see is that, yes, the mutant does regulate calcium differently. It's holding on to the calcium because it can't transport it. We see calcium in these trichomes, just about every map, and we never managed to catch any waves of calcium in the normal wild type leaves. So this is what we would like to do, and we're writing a grant right now to try and get this funded. We really need a complete change in how we image plants at the synchrotron. We need it to be much, much faster. We need to, and this is what we're trying to develop with Ryan Tapero. Anyone who knows his beamline knows he specializes in these custom sample environments for mounting specimens in front of the beam. We need to be able to image living plants. We need to germinate them in controlled environment in sort of modules that can be moved right in front of the beam. So it only has to be big enough to grow a plant to about the 2-week stage, which is where the majority of experiments done in Arabidopsis happen around this time. It needs to be a kinematic mount. It needs to go straight into position so that the entire floret is in the field of view. And then we would select the leaf we want to image using the XY stages rather than cutting a leaf off a plant, because we just can't risk introducing that abiotic stress. We need flexible raster directions in our scanning. We need to be able to draw a loop around the image and as quickly as possible and scan in whatever direction we need to scan in. When we collected some of the images I showed you at Brookhaven, we were, I think it was a 50 millisecond dwell time per point. We need to be at least at the most 5, possibly as shorter than that. We need to image as fast as possible, really so that we can reduce any kind of beam damage to the plant. I mean, I understand that, you know, synchrotron beam is many times hotter than the sun, but plants are adapted to being in the sun, and 5 milliseconds per point might be a controllable level of beam damage that we could actually consider repeat imaging if necessary. So that's what we're proposing to do. Our experiments have kind of helped us understand a lot about what's going on in the caxes, but we are still working with still images, and we need moving images. And to us, when we're asked to consider what we do with a faster beam or a smaller beam, then that's the main use of it, is we need to image fast enough that we can image like in a minute or so over and over and over again as these biological fast transient phenotypes are taking place in living tissues. So this is my APS upgrade wish list. So, you know, you've got an entire year to realize my wish list. And that's basically what I've been saying, order of magnitude faster analysis, so that we can do repeat imaging on living tissues. I would really love to see custom sample environments and mounts that can accommodate living tissues. So that we can image plants, image living plants instead of dead or dying or sectioned or preserved tissue. Because as we get down to really small beams, we're going to be just imaging sort of the damage that we've done post-mortem or in sample preparation. And I think our move to in vivo imaging is something, you know, that's very important to science. One of the other things that I would love to see in an upgrade is huge upgrades to the software, especially interoperable file formats, so that we can take that data away and look at it in other elemental imaging applications. I already mentioned I would really love to see flexible scanning directions. We shouldn't be manipulating the plant if we have a machine that can, rotate a stage and move X and Y. The less we handle our tissues, the better. And then loop selection of mapping areas so that we just reduce that time overhead of mapping nothing, mapping the corners and fresh air. We really need to get rid of that. That's an easy fix that we can get rid of the time we spend doing that. And then faster map setup. For our experiments, we can't spend an hour setting up a map. If we detached a leaf and we're going to get imaging it, it can't be sitting there for an hour before we actually hit start. It needs to be really fast and try and capture things that otherwise we couldn't capture. So that's all I've got. I'm happy to take any questions from anyone. I don't know if I came in on time, but
Almost.
almost. I try not to go over. So that's all I've got.
So I have a question to you. So what would be the resolution you can tolerate for the whole leaf imaging?
Well, we went pretty low on the laser ablation. We were at, I think, 30, somewhere between 30 and 50, the size of the beam, and then scanning at 300 microns per second. So on a leaf that big, we need to get the whole leaf because there's a flushing and there's various things going on. We don't really need to be at a fine level of detail. So we could really sacrifice that easily in terms of speed. If we can just show that signal moving, then we would achieve our aims.
Thank you. So no, it is very interesting project, like pushing the time resolution of microscopy techniques. Yeah.
Yeah, I mean, we would like to get towards like elemental movies rather than still images, and that is going to take a lot of sample, custom sample environments, design and iteration. So that's what we'd like to see, and then we'd be absolutely love to come back to the APS to do this.
Yeah. Okay. Thank you. And if we don't have any other questions from the audience, so I'd like to move. Oh, there's something in the chat.
Yeah, what extra energy are you collecting? I think it was probably 10 keV or 11 keV. The calcium concentration is like in the weight percent. It's like 12 weight percent calcium. It's tens of thousands of ppm. So yes, 5 millisecond dwells might not get enough counts. Yes, so that is a risk. I think with calcium we're doing well because it's just so enriched. That we might have a chance. I mean, obviously we were doing arsenic or cadmium. I don't think we'd see anything, but for something that's abundant, maybe we can start to see at 5 milliseconds. But those are great questions. Thank you, Jocelyn Richardson.
Okay, so now I would like to move on to our next speaker. So you probably heard me saying laminography like 5 or 10 times during my introduction, and we're wondering what is that. So Victor will explain what it is. It's our new technique coming to APS and being available after upgrade at multiple beamlines for biological and environmental science samples. So Victor received his bachelor and master's degrees in information technologies in 2011 and 2013 respectively from Novosibirsk State University in Russia. He has PhD in mathematics from Lund University in Sweden. Before joining Argonne in 2020, right before lockdown, he was a postdoctoral researcher at Max IV lab in Sweden. So Victor is our BM tomography expert, making it faster, more efficient, with more efficient and fast reconstructions. He is interested in inverse problems in tomography and tachography, high performance computing with multi-GPU systems, and conducting complex dynamics X-ray experiments. So Victor, please go ahead.
Right. Thanks for introduction, and I hope you see my presentation. Could you confirm?
Yes.
Okay, thanks. Yeah, again, so I'm Victor. So I'm a beamline scientist, just a beamline scientist. I'm not a biologist. So that's why at the beginning, I just want to say that I apologize for any problems with terminology in this presentation. And today I want to talk about this new technique called laminography. So this is my outline of the presentation. So first I will show which beamlines our imaging group has and will have after the APS upgrade. Then we will talk about what is laminography, discuss advantages and disadvantages of this technique. Then we talk a little bit about how we do reconstruction for this type of technique. And then I show some biological applications, and then we look at conclusions and outlook. Okay, so you already seen this slide today. So these are our imaging group beamlines before and after the APS upgrade. So you see that we cover the whole resolution range, starting from 10 nanometer and up to 10 micrometers. So for that we have 3 instruments. So one instrument is this micro CT beamline at 2 BM, where we work with 1 micron resolution. After the APS upgrade, imaging at this beamline will be much faster. And we also plan to do some laser cutter for the fly sample preparation at this beamline. So again, in these notes here in black color, I show what we have currently or what we had like before the shutdown. And in blue color, I show what we will have after the APS upgrade. So again, so before the shutdown, we also have the TXM instrument, transmission X-ray microscope, where you're able to do ZENS techniques and work with energy 6 to 12 keV. After the APS upgrade, this instrument will be much faster. We expect like 10x speed gain. And we also want to do some dynamic in-situ experiments there. So besides 2BM and 32IDB, the TXM instrument, it will be in 32IDB station. We will have the projection X-ray microscope at 32IDC station. So this instrument, it will cover this gap of resolutions between 50 nanometer and 1 micron. And there we will do holotomography, like this phase contrast techniques. So it will be more efficient in terms of the deposition dose in the sample compared to the TXM instrument. And it will also allow us to study much thicker samples compared to the TXM instrument, where we're able to measure only 50 micron samples thick. Okay, so this is like overview. And as you see here, there is this laminography technique in each of these beamlines. So it is in black color for micro CT because you already implemented it. And we also plan to implement this technique in transmission X-ray microscope and with this new projection X-ray microscope. So, and what is this laminography? Yeah? So this is like a simple figure demonstrating what is laminography. So on the left side, you see regular tomography. So we put a sample on the stage, rotate the sample and collect projections. Yeah. And it becomes really problematic if your sample is really flat, yeah, like in here. So if the sample is really flat, then X-rays cannot propagate through this sample in this direction. And if you put your sample vertically, there will still be some blocked views for some range of angles. So this problem can be solved by laminography. So in laminography, what we do, we tilt the rotary stage, and the propagation distance for the sample will be much smaller all the time. So while we're rotating the sample. So in these green lines, I show how X-rays propagate through the sample. So it's quite huge distance in this first case. So then it will be also quite huge distance for some angles in the second case, but for laminography, this distance, so X-rays propagating through the sample is much smaller. So we have quite nice photon counts on the detector. So this is the benefit. So laminography is basically used for scanning these planar samples. It could be some leaves, some mouse brain slabs, and different types of samples. And we implemented this laminography at 2 BM, at the magnet beamline of the imaging group. So if you already conducted experiments at 2 BM, you remember that we have the rotary stage. So on the left side, we have the detector. So X-rays are coming from the right side. And for laminography, what we did, we just put a wedge under the stage. But under the stage, we also have the motor that goes between -10 and +10 degrees. So that's why we can quickly switch between two setups. Yeah. So if we put this tilt motor to -10 degrees, and then we add this 10 degrees wedge, so it gives us regular tomography. If you put this tilt motor to +10 degrees and +10 degrees wedge, it gives us 20 degrees, which is the laminography. So we can quickly switch between two setups. And this is on the right side, you see how we typically mount this laminography sample. So here is some flat sample of the size 300 micron by 4 millimeters by 4 millimeters. So we just take a Kapton tube and glue this sample to this tube. So since Kapton tube is transparent to X-rays, so we can do laminography easily. So this is somehow just really the easiest way to implement laminography. We don't need anything, we just put some wedge under the stage and measure our samples. Yeah. So, okay, let's talk about advantages and disadvantages of laminography. So the first advantage is that laminography prevents the beam blockage. So if you take like this flat sample, like in this case, this is some mouse brain, and look at the projection data on the detector for tomography. So you'll see this slab here, but we will not have any photon counts on the detector. So in here, black color means that we don't have any photons on the detector. So white color means that we get photons on the detector. So basically this color bar means transmission. Yeah. So when we do reconstruction, so we get attenuation. So somehow these colors are flipped. Yeah. So brighter color means that sample has higher attenuation, and darker color means that sample has lower attenuation. And then since the beam is blocked for this regular tomography, so then our reconstruction, of course, will be very bad. So you will not see any features inside the sample. Yeah. And you see, if we tilt this sample and we start to get some acceptable photon counts on the detector, and then of course we can do nice reconstruction. So features can be segmented here. So this is the first advantage. So it prevents the beam blockage. So second advantage is that we have less local tomography artifacts and less sample damage. So if we have a really big sample and our detector has like really small field of view, so we have a really small region of interest. So in order to reconstruct this small region of the sample, you still need to propagate X-rays through the whole sample. Yeah. So these X-rays propagate through the whole sample length, and then they are captured by the detector. And the problem here is that, of course, we damage the whole sample this way, because X-rays propagate through the whole sample. And then we also get some information from the regions that we don't want to have. Yeah, like from here or from here. And of course, this affects the reconstruction quality. So in laminography, X-rays propagate, and this problem, we call it local tomography artifacts. Yeah. So in laminography, we decrease the amount of these local tomography artifacts because X-rays propagate not through the whole sample, but just from a part that is much smaller than in tomography. Yeah. And of course, in this case, we don't damage the sample on the border, for instance, here and here. So this is the second advantage. So the third advantage is that laminography is more optimal for scanning large samples. So if you have a really huge sample, like, I don't know, monkey brain or human brain, you still need to cut it somehow, yeah, to get nice photon counts on the detector, to have appropriate transmission for the sample, yeah. So for tomography, you need to cut this sample into pillars like this on the left side. And then you need to do like a vertical scan of the sample, yeah. So for laminography, you can cut this sample into slabs like this, and then scan these slabs, yeah. So you can move the motors on the stage and scan different parts of these slabs, yeah. So this is another advantage. So we can significantly decrease the number of cutting procedures. Okay, so next is advantage is easier sample preparation for nano-CT type techniques. So if you've done experiment at 32ID with the transmission X-ray microscope, you probably remember that it is always difficult to mount your samples. So we mount samples on the tip of the pin. This is like good sample preparation on the right side. You see the sample size is 50 micron, and it's very difficult to put your sample on the tip of the pin. So if you put your sample somewhere on the border of the pin or somewhere else, yeah, so it will be really difficult to scan, and it's not difficult, it's not possible to scan this sample in tomography setup. So this is like bad sample preparation. So if we implement laminography technique for nano CT, we can put our samples on the Kapton tape that could be like 1 millimeter by 1 millimeter size. So we can put our samples like a powder on this tape and then scan different particles in different regions. Yeah, so it's much easier sample preparation than mounting one particle on the tip of the pin. Okay, so these few advantages. So one disadvantage is that we have start to have undersampling artifacts. So here I show some synthetic data examples when we do reconstruction for different laminography tilt angles. So we have reconstruction, and this is like a vertical slice for a sample. So for if we have laminography angle 0, this means that we do regular tomography, so we don't have artifacts. Yeah. If we do laminography angle 10 degrees, so we start to see artifacts like this. So these are these undersampling artifacts. And then if we increase the laminography angle like 40, 50, 60 degrees, then the quality becomes really bad. Yeah. But this stuff can be partially addressed with iterative schemes that I'm working on now. Yeah. Okay, so this is a disadvantage of using laminography, but you can still see that for small angles like 10, 20, 30 degrees, the quality is still okay. Okay, let's talk a little bit about laminographic reconstruction. So I will not go to the details here. So just say that we use this TomoCuPy package for that. So laminography reconstruction. So this package allows to do tomography and laminography reconstructions on very huge volumes. So it is accelerated with graphical processor units. So it is 10-30 times faster than TomoPy. So in this, we always do reconstruction at 2 p.m. and 32 ID by using this package. And yeah, and basically reconstruction in this case is faster than data acquisition. Yeah. Yeah, I just want to show how we use this TomoCuPy CLI. So CLI means command line interface. So we do reconstruction this way. If you know the rotation axis and laminography angle, so let's say your rotation axis is 1200 and laminography angle is 20. So then you just write TomoCuPy recon and then you put the file name and then you put rotation axis and lamino angle. Then you get this reconstruction. Yeah. So so if we don't know the rotation axis, so we do this manual search. So we put the reconstruction type try and basically reconstruct our sample with different rotation axis. And then you see that if the rotation axis is off, we see the artifacts. And then we basically slide through different images and select the image with best quality and see to which rotation axis this image is associated to. Yeah. So, and these are general steps for regular tomography, but for laminography, it is also makes sense to check the laminography angle. So in our case, we put some wedge under the stage, but we don't know exactly what is the angle of this stage. Yeah. So that's why also in TomoCuPy, you have the option to search the laminography angle. Yeah. So if laminography angle is a little bit off, then you start to see artifacts on the borders of the image, like this. And again, so you scroll through all images and see that the best and select the best one. Yeah. So the procedure, the whole reconstruction procedure is as follows. So you start with searching the rotation axis and check the rotation axis in the middle of the image. Then you check the laminography angle. You found by using the found rotation axis, and then you basically find this laminography angle and put this laminography angle and do the full reconstruction. So this is how we do reconstruction of laminography data at 2 p.m. Okay, so and again, this reconstruction is really fast. It is implemented on GPU. It is much faster than data collection. Okay, now we will talk about applications. Probably most interesting part here. So I already showed you these mouse brain slabs. So here you see the slabs. So they're like 1 centimeter in diameter, and thickness is about 1 millimeter. So the thing is that for tomography, you can't get any good results because these slabs really fully block the beam. And the only way to scan the sample is by using laminography. Yeah, and these are the results here. Yeah. Okay, so next example is this leaf, devil's ivy. So this leaf was taken basically. So this is a plant that grew near Francesco's office. So he said that he brought this plant from Italy, and yeah, just took 1 leaf of that to scan. Okay, so you see that we can scan really huge objects in laminography. Yeah, so this leaf is quite huge. This is what happens with the leaf if you scan it with tomography, with regular tomography. So you see there are some beam damage of the leaf in different directions. And this damage appeared only with tomography. So when I scan this leaf in laminography settings, so the damage was only in the middle. When I scan it in tomography, so I start to have problems also on the borders. So here you see the reconstruction. So we go through slices. So you see some bright lines here. I guess you should understand what is it. It's, you know, biologists. So with the region I scanned here is the region near the stem. And these are like basically parts of the leaves coming to this stem, and there are some bright stuff there, high density stuff that I hope you know what is it. And okay, I'm talking about this bright thing here. Okay, this was the leaf, and I also compared the quality, reconstruction quality between tomography and laminography. Again, so here we have this local tomography problem, so that for reconstruction we use parts of the leaf, parts of the data that is not related to the middle region. So that's why we start to see really huge noise and these local tomography artifacts. And again, we have higher noise, and then we also have sample damage. So we clearly see advantage of using laminography in this case. So the signal to noise ratio is better and sample is not damaged. So another sample is this butterfly. So we scan it like before the dark time with Olga. So this is like the wing of the butterfly, and this is Olga preparing the sample. So we scan this sample, and these are reconstructions. So these are some parts, I don't know, but again, so I'm not very good in terminology. This is some parts inside the wing that has high density. And these are some examples of reconstructions. So I just want to say that the range of applications of laminography is quite huge here. So we can also scan different insects. Okay, so then this is the last application I show you. So this is characterization of mouse bone treated with hydroxyapatite spheres. And here, yes, so we scan this sample in 2BM. Here, what we try to see these small features. So this is how basically the bone grows and basically recovers with using these spheres. And to compare, I also compared this sample for scans for tomography and laminography, and then you also see that these small particles here, these small features, they are not visible in tomography, so it's very difficult to segment anything here between these spheres. And but laminography gives quite nice results. And besides, we also scanned this sample with XRF. So this was like the first experiment to IDE made by Olga with laminography. So this is the absorption contrast, pixel size 20 micron, and this is like XRF computed laminography where we looked at the calcium. So this was the first experiment. I hope after the upgrade we will get much higher resolution with XRF. Okay, so, and I will go to conclusions and outlook. So again, I just say that laminography has many advantages. So prevents beam blockage, less tomography, local tomography artifacts, reduces radiation damage, reduces the number of cutting of larger samples. And of course, it's easier sample preparation for nano-CT. So the disadvantage is this undersampling artifacts that I hope can be handled by iterative schemes. So we already have software for reconstruction in TomoCuPy, so one can welcome to use it. So it's quite easy to implement laminography at any micro CT beamline, just put wedge under the stage, but it is of course much more difficult for nano CT because there are different stability issues, and then stiffness, etc. But it should be still possible to do, so that's why it's also in our plans. So we already tried this technique for different samples at 2 BM. So in biology, material science, energy materials, and our stuff, yeah. So in our outlook, what we have is that we want to implement this laminography at 32 ID nano-CT instruments for this TXM and PXM that we are going to have after the APS upgrade. So we're also working with Olga on implementing XRF laminography with 2 IDE. And they're also joint work with 8 ID beamline, where we do coherent surface scattering imaging, John. So basically, yeah, I believe that laminography will become very popular after the APS upgrade. And yeah, I also plan to accelerate all these iterative schemes with using supercomputers. And yeah, just I'm welcome. You're also welcome to try any laminography imaging after the APS upgrade at all our beamlines. Yeah, thank you for your attention.
Thank you, Victor. Does anyone have questions to our last speaker? Or maybe
I had a question. The Victor, very nice. That I was wondering, when you said that for the Kapton tape samples in between the Kapton tape, you still need to cut it down into that 1 millimeter by 1 millimeter, or it can be a little bit larger.
Yeah, but I think it's not difficult to cut the sample to 1 by 1 millimeter, but it can be larger. So I'm not sure how much larger it could be, because we will start to have these stability issues, you know, in nano CT. Yeah, when any wind around the sample may basically affect the image quality, and then if we have something which is like 5 by 5 millimeter, so it will be not very stable. I would still try to make it much smaller. Yeah.
All right. Thank you.
Do we have any questions to our speakers in this session? Maybe somebody didn't have a chance to ask the question before. So yeah, then I'd like to thank all our speakers of today. Thank you for your great talks. I think we're all getting inspired. And now getting sad that Beam is not available for next 12 or so months. So thank you, everybody. And if we don't have any questions, I would like to say goodbye, and I see everybody tomorrow morning for our next session of the workshop.
Yeah.
Thank you.
Bye.